Good afternoon, ladies and gentlemen, and welcome to this official opening to the 7th International Scientific Digital Symposium of the Ordesa Cátedra. Ms. Cristina Campoy is accompanying me in this panel, director of the Cátedra, and Ms. Pilar Aranda, rector at the University of Granada. First of all, on behalf of Laboratorios Ordesa, I would like to express my support and all my condolences to those people suffering the direct consequences of the COVID pandemic, and also our congratulations to the clinicians, to the healthcare professionals that made the greatest efforts to mitigate the serious consequences of the pandemic. It is a pleasure for me to welcome you all to this 7th International Scientific Digital Symposium. And due to the current circumstances, we will hold it remotely today and tomorrow, and the general title is Early Nutrition and Reprogramming of Health and Disease. As you all know, fostered by Laboratorios Ordesa, the Cátedra Ordesa for Child Nutrition was founded in 2013 together with the universities of Zaragoza and Cantabria, and later on the University of Granada adhered to it, and more recently the Santiago de Compostela University in the northwest of Spain. This is the first inter-university cátedra professorship in Spain that, well, groups 4 different universities, which increases the research potential. Its main goal is to foster research on child nutrition and to train healthcare professionals in this area. Undoubtedly, this will be a highly interesting symposium in that sense. I would also like to express my gratitude to all of you for attending remotely to this 7th International Symposium. The participation of so many professionals coming from the pediatric sphere from all over the world, just for your information, more than 1,400 people registered already from Asia, Africa, Latin America, and Europe. So thanks to your presence, your attendance, we will be able to enrich the scientific program, which was designed by the Cátedra. I would like to thank the speakers and participants of the different panels for their willingness to participate, to take part on these different roundtables, and for being willing to share their knowledge, their expertise because they're valuable and top level presentations will provide to us, to our company knowledge to keep on progressing in the quality of children's nutrition. These are the pillars of our company to put at your disposal, to the disposal of the society as a whole, better and more secure and safer foodstuffs for infants. Last but not least, I would like to thank the members of the Cátedra for their work in preparing this remote symposium, and to the technical team that made this possible. More than 2,300 attendants are following this symposium. So I wish you all a very fruitful symposium. Later on, now I'm going to give the floor to Ms. Cristina Campoy. She's the director of the Cátedra Ordesa. I thank her for her personal involvement and the effort she made to be present today, and the great task she's performing. She's carrying in this. Thank you very much, Shukran, and many other languages of the different attendants we have today with us. Thank you. Good afternoon, everybody. Thank you for being here. First of all, I'd like to say it's a real pleasure to me. It's a real pleasure, and I'm honored to have you all here. Well, this pandemic started a year ago, and it changed our schedules, our everyday lives However, the possibility of holding the 7th International Scientific Digital Symposium for the first time remotely, it's, well, just some fresh air and a step forward in the normality, in the coming back to normality we all aspire to. I would like to thank all those people that are suffering important sequels as a consequence of this pandemic. Since I accessed the cátedra as a director, I try to lead it towards internationalization with the main aim of achieving the main aims by progressing, advancing, and transferring knowledge to all professionals involved in the field of child nutrition. I feel happy to say that, well, the steps we took made us come closer to reaching those objectives. All the things we've done wouldn't have been possible with the collaboration of the members of the mixed committee, composed of the people that I'm showing here, belonging to the 4 different universities, Zaragoza, Granada, Santiago de Compostela, and Granada, and also the expert from Ordesa Laboratory. Cátedra Ordesa is well known all over the world nowadays, and thanks to her excellence, she is a leading entity. So the rector of the University of the Granada will talk to us today. She will address us, and I thank her beforehand for her support to the activities organized by the Cátedra Ordesa, and on my behalf, welcome to this symposium. You have the floor, Ms. Pilar Aranda. Thank you so much, Professor Campoy. Thank you so much, Joan. I feel absolutely happy and deeply honored to be able to share with you these minutes. I feel especially happy to be here with Professor Campoy. She's a human being that has devoted her soul, her body to research. And with her effort, she had a master's, she organized a master's course and a PhD. And back in 2015, I got to know this cátedra, and I had the opportunity to share important times with my dear friend Montserrat Rivero, and I've seen it growing little by little, extending it to the recent incorporation of Santiago de Compostela University. I would like to thank Ordesa for her deep commitment with research. Public and private partnerships are more and more necessary considering the current scenarios to be able to support all the activities launched and fostered from universities. And society plays a really leading role, and I think it does a good thing in demanding more and more research. Cátedra Ordesa is well known not only for its track record in quantitative terms, but also in qualitative terms. Working in the field of nutrition, of child feeding, I was trained with Professor Matthijs, and I inherited from him, I got from him, from Professor Valela, that passion, that love for research, and especially in these fields. So the outcomes and the research we're obtaining, the findings are absolutely outstanding. And it is absolutely necessary, indispensable to speak about child nutrition, because this has a direct impact on reprogramming. That is the initial impact of nutrients on the development of the central nervous system and on other vital organs. They have a persistent impact on throughout the lifespan, span, homeostasis, stability, etc., etc. I've been able to exchange many data about the importance of fetal reprogramming, the importance of maternal feeding, the role played by the placenta as a regulating organ, and over to the moment of delivery, etc., etc. So I would like to remind all of you of the great efforts made by the different clinicians, by the different professionals. We are obliged to do so. The first social shame for all of us is that people are dying still in our days. On the 21st century, there are children that dying of out of hunger, famines, etc. So thanks to the commitments, the social responsibility of Ordesa, there are exceptional programs that try to socialize, to democratize knowledge, and also to cater for different emergencies that take place in so many situations all over the world. This COVID-19 pandemic brought about two advantages. It has two virtues, and this is an important reminder. And of course, my condolences to the relatives of people that left us due to this terrible disease. So, thanks to the pandemic, we are able to be in contact in real time with, right now, nearly 700 people. Before that, in a face-to-face modality would be much more difficult, despite the fact that we still miss presentiality, face-to-face, of course, a lot. But despite everything, the pandemic made us feel humble, that made us humble. We are vulnerable. We need strong healthcare services. We need research. Within some months, we've got vaccinations. In other circumstances, years would have elapsed. And this was based on basic research, on basic investigation. That was the result of public-private partnerships, always at the cutting edge of research, such as the Ordesa Cátedra, making an effort so that society improves. Without science, there is no future. And I dare say, without public-private collaboration, partnership, there is no future. So again, thanks again to Ordesa Cátedra. We've been collaborating for many years, and I'm sure they will continue. They will maintain this this strong commitment. They keep on organizing events. And please allow me to finish with a congratulation by congratulating Ms. Cristina Campoy, Professor Campoy, for her capacity to involve us all in projects that make us feel enthusiastic, and I feel really proud of having her as a friend, as a colleague. Congratulations to all speakers, an exceptional and extraordinary program agenda, and I will be following closely all the findings, all the conclusions, because they are in line with my research interest. You are at the cutting edge of knowledge, so we need you all. I wish you a fruitful symposium. Thank you. Thank you so much, Pilar, Ms. Professor Aranda, for participating in this official opening. And without further ado, I'm going to start to open the first panel, the first round table. In this case, I will co-chair it with Dr. José Antonio Moreno. He works at Laboratorios Ordesa. We've known each other for many, many years. He's an outstanding scientific. And without further ado, I'm going to present the first panel. As you can see in the program, today's agenda, it will be about microbiome, development of the immune system within the two first years of life during infancy. So we welcome Professor John Cryan, Dr. Carmen Collado, and Dr. Chris, and myself. First of all, I would like to introduce Professor John Cryan. He's, well known all over the world. He's Vice President for Investigation and Innovation at the Cork University in Ireland. He's also Principal in investigators at the Microbiome Institute, and he has held different positions, and he has stayed at different departments, psychiatry in Australia, postdoctoral stays at Pennsylvania, Philadelphia, United States, and also in California. And moreover, he's been awarded with many international awards. Apart from that, regarding his current research, he's focused on studying the gut-brain axis, macrobiota brain axis, how this impacts on stress and also in immune disorders. So we can see the different time windows across the lifespan to prevent disease. Apart from that, Professor Cryan has published more than 550 peer-reviewed articles. He's a senior editor of neuropharmacology and stress neurobiology, and he belongs to the editorial board of more than 15 journals. I would like to add that he's published a very well-known book, a bestseller, on the psychobiotic revolution. And currently, as I said, he has received numerous awards from all over the world. He was a TED-Med speaker in Washington, and he's past president of the European Behavioral Pharmacology Society. So, in a nutshell, he is a top-notch speaker who will speak to us about preparing for life, early nutrition, and the gut microbiota brain axis. You have the floor. I think we have to introduce to the two speakers. Yes, if you don't mind. Sorry, Jose Antonio. Yes. Hello. I will introduce the other two speakers of this panel. So I have the pleasure of introducing Ms. Carmen Collado. She is a researcher at the Institute of Agrochemistry and Food Technology, IATA, here in Valencia, Spain, at the Spanish National Research Council. Apart from that, Ms. Mari Carmen Collado has been a adjunct professor since 2007 at the University of Turku in Finland. From her first postdoctoral, she's worked in the field of probiotics microbiota, the relationship between the infant and the maternal microbiotas, how they interact, which are interactions between breast milk and the infant microbiota. And so we will get to know very interesting knowledge about the links between both macrobiotas and the important role microbiota plays in the development of the child throughout the lifespan. And thirdly, Mr. Antonio Suarez, he holds a chair at the Department of Biochemistry and Molecular Biology at the Pharmacy School of the University of Granada. He's been working in the field of microbiota for a long time. He has several publications in that sense, and he has organized a very interesting team in the field of bioinformatics of gut macrobiota. He's studied on functional and taxonomical dynamics of gut macrobiota in human health and disease, and he will speak about his last findings, his last studies or research. It's a publication that will come out shortly. Thank you all for listening. I remind you that on the lower bar, you can ask your own questions through the chat. Please write them down. And you can also choose the simultaneous interpretation. You can choose between Spanish and English. Thank you so much. Without further ado, Professor Cryan, when you're ready.
Thank you very much. I'm delighted to be here. And I'm going to talk to you today about priming for life and how the early life nutrition really affects the microbiome gut-brain axis. So I work in a medical school, and in medicine, everything goes back to Hippocrates. And Hippocrates is thought to have said, now, it could be fake news, but he's thought to have said that all diseases begin in the gut. And, you know, that can be quite strange when I talk to a lot of my friends in areas of psychiatry and neurology and brain development. But all we have to do is really just look to our language and see how we personify our emotions in our gut. We have gut feelings, we have gut instincts, we feel gutted when we're disappointed, we make gutsy moves when we're brave, we have butterflies in our tummy when we are nervous. So, you know, perhaps there is an underlying biology to these well-worn phrases. Now, the concept of gut-brain signaling, you know, dates back to you know, the times of Pavlov and Darwin, well over 200 years, and is well studied in the context of food intake. And people understand, you know, that you know, your gut talks to your brain to tell you to eat and when to stop eating. But more recently, in the last decade or so, there's been a greater appreciation about the role of the microbes in the gut in regulating the signaling. And that might at first seem weird, that you would want to focus on the microbes if you're interested in the brain. But really what I do need to remind you is that we are living in a microbial world. Microbes were here long before humans, and thus perhaps it's not so surprising that we have co-evolved with these friends, and these are friends with benefits. And so if we just look at some of the numbers that we have in terms of the microbes, for example, we have more than in our genes, we are more than 99% microbial. Very humbling. The weight of our gut microbes are roughly that of our brain. And if we looked at the number of cells we have more microbial cells than we do human cells. So that's really humbling as well. And with this field, well, really the two big challenges that we face, and this is what our group is really trying to understand, is how can microbes in the lumen of the gut send signals to the brain to affect emotion and behavior? What are the pathways of communication that go? And so we're slowly beginning to disentangle this, although we have still some ways to go overall. And most of this work is being carried out in animal models. And the next part of the puzzle is, how do we go from animals to humans? And that's also informing a lot of our work right now. And it's not just the microbes that are important, but it's what they're doing. So one of the big areas right now is trying to understand what these microbes are doing. And these microbes are like little factories producing all kinds of weird and wonderful chemicals that our bodies wouldn't be able to have otherwise. Among these are chemicals like short-chain fatty acids, for example. And so a lot of the focus in the research is trying to say, well, how do we get the raw materials, which is from nutrition and the diet? How do we, you know, bring them into the factory, and then they're acted on by the workers, which are the microbes, and then to change the pathways of communication to the brain. So if we want to modulate this, we can either change the workers, but change management in workplace is difficult, and that is the same with the microbes that are there. Or we can change the nutrition, the raw materials, maybe start with something a little different, give them a heads up about what we should be doing. So that's kind of where the field is really going, and I'll share with you some of the work that we've been doing in that sphere. And perhaps the greatest example of this is really in human breast milk. Human breast milk contains the highest complexity of sugars compared to any other mammal. And evolution doesn't do that without some reason. And it was really surprising to me many years ago when I realized that these sugars, they can't even be broken down by the infant. It has no capacity. So that's not evolution working. So what's going on? Well, what's going on is that we've kind of given up that job. We've outsourced it to our microbes. And the microbes, therefore, break down these human milk oligosaccharides to key chemicals like sialic acid that are very important for brain development. So you can see that this coevolution of the microbiome with our nutritional environment is really important. And so this is really important throughout the lifespan and throughout early life in particular. And so there's a growing appreciation of what are the factors that influence the early life microbiome, whether it's exposure to antibiotics, to the environment, to infection, different dietary habits, various all of these can influence both the mom's microbes and the infant's microbes. Now, we get our microbes, our early microbes, kind of like a birthday present from our mothers on the day of birth. We're taught mostly to be sterile in utero. But that doesn't mean that what happens to the mom during pregnancy or even preconception, is more focus on that now, could influence the mom's microbes, both the gut microbes and the vaginal microbes that are handed off in this kind of evolutionary relay race overall. So a lot of these factors can influence the mom and then affect what type of microbes that are handed over. And then we also know nutrition during early life, whether, especially in relation to mode of nutritional provision and solid food introduction, are really important aspects of shaping that early life microbiome in general. And so we've written about it in this article that's just out, which was sponsored by Ordesa, to really highlight where the different nodes within early life that the microbiome can be shaped by nutritional influences. And here we try to highlight how changes in nutrition during this period can impact the different pathways of communication between the gut, through to the enteric nervous system, through to the immune system, up through the vagus nerve, or independent of vagus, but onto the brain to change brain function and behavior. And also, another factor that we've been really interested in early life in shaping the microbiome has been mode of delivery at birth. And this goes back to work from Gloria Domingos-Bello some years ago, which showed very clearly that the microbiome of infants born by C-section is very different to that born vaginally. You can see here in blue is the cesarean and in pink the vaginally born. And that was a small study. It's been reproduced now in at least 14 or 15 different large cohort studies, most notably the TEDdy study, which highlights here that birth mode is one of the key factors, as well as mode of nutritional provision, with breastfeeding also having a significant effect on the microbiome in early life. And you know, what we're beginning to see, that this microbiome is really a security blanket during early life in shaping and programming the body's systems in general. But it's not just about missing microbes, which are really important, but it's also, we need to point out that it's the blooming of pathogenic or dysbiotic microbiome overall. And so, as can be seen in this work here from Trevor Lawley's group. So we're beginning to really tease this apart more and more. And we know that birth by C-section leads to a greater risk of allergy and asthma and type 1 diabetes, but we know less about it in the context of brain health, what I'm interested in. So some years ago now, and just published in the last month, work from Livia Mores, who's now at Caltech, in my group, showed that we developed an animal model of C-section. And in these animals, we could see that there were deficits in their microbiome as very early in life. In particular, we found there was reductions in bifidobacteria. And when we looked at their behavior as early as postnatal day 9, we could see changes in behavior, but all the way through at adulthood, there's this enduring effects on behavior in terms of social behavior and anxiety behavior in particular. So we wanted to go one step further and see, could we rescue this? Could we, by intervening in early life, change this? Now, mice, like some mammals, are coprophagic in that they eat each other's poo. So we took advantage of this and basically performed a fecal transplant, basically housing the vaginally born with the C-section animals to see could they basically by artificially fecally transplanting restore the effects, and indeed we could, which is really exciting. And then but that's not amenable to humans perhaps, so then we wanted to go one step further and then look at what could we put bifidobacteria back. And so we treated with a bifidobacteria breve, and we were able to reverse the effects, as well as with the prebiotic, which stimulates the beneficial bacteria. So what we're beginning to see is that yes, in C-section, there is deficits in the microbiome gut-brain axis, but the good news is that perhaps we can look at nutrition as an intervention strategy to try and reprogram or reverse some of these effects overall. And this is something that we're very excited about, and we feel that because C-section, although the rates are going up perhaps more than they should, it is still a medically indicated life-saving intervention, and we would like to be able to offer to moms and families the opportunity to have intervention strategies that could counter the negative impacts of C-section on the microbiome in early life in general. Okay, so that's a lot of our work, and we've been really exciting, but my neuroscience colleagues, they're a skeptical bunch. They need more evidence. How do you to really implicate the microbiome in brain function and development? And so what we look to is other areas of science, and like engineering and biology even, where we want to get evidence that something is important. Well, you take it out and you see what happens. And so the concept of being germ-free, well, that's not, that goes back to Pasteur's time. And here you can see a germ-free man from a sci-fi magazine from the 1920s. Now, our germ-free mice, while they have their own limitations, have been very useful to allow us to answer the question, is the microbiome involved, yes or no? However, they don't really have any human translation. I mean, the exception being the boy in the bubble that Paul Simon sang about in the 1980s. And we have worked with these models over many years now, and really they've been transformative in our thinking in terms of being able to show that if you don't have microbes in this extreme situation in early life, the brain doesn't develop properly. And that's really key. And we've shown that, for example, myelination patterns, myelin is the insulation that's really important for brain conductivity, that the myelination patterns are dysregulated in the prefrontal cortex, a area of the brain involved in higher cognitive function. The regulation of adult hippocampal neurogenesis, the birth of new neurons, is out of kilter, and this is very much a process involved in higher cognitive function and pattern separation as well. A variety of changes in neurotransmitters, and more recently we've shown that synaptic plasticity doesn't function appropriately in these germ-free animals, as well as changes at the immune cells of the brain, the microglia, and at the level of the blood-brain barrier. So very exciting. And here you can see, you don't need to be a neuroscientist to see that this is a neuron from the amygdala region of the brain involved in fear learning, involved in pain processing, involved in social behavior. And you can see that it's almost like in a stress state if the microbes are not sending signals to the brain. So what does this translate to at a behavioral level? Well, at a behavioral level, it translates to deficits in fear learning, as we showed in this study, deficits in visceral pain. And visceral pain is a core symptom of adult diseases like irritable bowel syndrome, but also has very clear overlaps with infantile colic. And there may be a relationship between the microbiome, gut-brain axis, and colic generation, and how nutrition could be intervening in that is really important. Also, in terms of sociability, we find that mice, like humans, are quite social, but if you give them the chance to spend time with another mouse, they'll gravitate towards the mouse, but not if they're germ-free. And mice, well, like some humans, can be a bit fickle. So if you get them to spend time with a new mouse versus their everyday mouse, they'll prefer to spend time with the new mouse, but again, not if they're germ-free. And so we're finding that there are clear aspects of the social brain and social brain development that are gone awry without these microbial signals overall. In relation to germ-free being an extreme model, we've also started looking at other models, such as early life antibiotic exposure. This is a study in rats some years ago with Siobhan O'Mahony, where we showed that giving vancomycin in early life left an enduring effect on visceral pain processing. And that was kind of remarkable, showing that that even though the microbiome recovers quite quickly, as you can see on the top left here, that there are these scars of early life microbiome effects on behavior. And more recently, we've been looking at this in breaking down other antibiotic exposures that are perhaps more clinically relevant. And we looked at penicillins versus a cocktail, and we find that there are deficits in anxiety behavior, in social deficits overall. And there's something about the social brain, time and time again, that we see that makes it sensitive to what's going on. And we have examined this across the entire animal kingdom, and you start to see the data is patchy, but you start to see when you interfere with the microbiome, you can change social hierarchies and social structures. And then when you change social dynamics, you can also change the microbiome. And this is, very important in this time of extreme hygiene that we have and social isolation in terms of lockdowns, and what that's doing to nutrition. And so we're beginning to see that we can juxtapose what's going on in the brain with what's going on in the microbiome, and that there are critical windows in early life in particular. And we need to understand how this plays out and how this leads to changes in executive function and other higher cognitive functions overall. And also we're beginning to realize from a developmental origins of brain health perspective that the microbiome is another pillar added to both the prenatal and postnatal environment and host genetics overall. We've also been working on showing that early life trauma and early life stress, and this is data from well over a decade ago now, where we've shown that adversity in early life leads to whole body syndrome, but among this is changes in the microbiome, and this has also been shown in humans. And so the next point is, can we reverse this? And we've been working to try and do this with a variety of strategies, nutritional strategies, to reverse the impact of this early life stress on the microbiome. And here you can see some of our efforts looking at specific strains of bacteria, Lactobacillus rhamnosus, alone and in combination with prebiotics or some of its soluble mediators that are having an effect, and also looking at milk fat globule membrane and prebiotics on this. And then other approaches, nutritional approaches we've been looking at include polyunsaturated fatty acids, and more recently polyphenols, which are present in a lot of dietary substances like nuts and a variety of vegetables as well, giving a lot of color where there's a lot of color. And these flavonoids and non-flavonoids are really interesting and potentially also in early life in shaping the microbiome gut-brain axis. We've also been focusing on adolescence and the impact that nutrition, and in this case bad nutrition, can have on the microbiome and have enduring effects there. And then what we can do to kind of counter stress effects in adolescence. And this was a study together with Beatrice Pisani's group where we're able to show that supplementation with vitamin A and polyunsaturated fatty acids could reverse the effects of stress. We've also been working on antibiotic exposure during this critical period, and we feel this adolescence is an untapped area for nutritional interventions on the microbiome gut-brain axis. This brings us to the concept of psychobiotics. It's a term we've coined here in Cork, and we really need to move these animal studies into the human domain, and it means targeting the microbiome for mental health benefits. We've talked about the different approaches that we can do that. We can use probiotics, prebiotics, symbiotics. Does the bacteria have to be alive? Could we use a paraprobiotic? And then fermented foods, and we have a lot of exciting work in the fermented foods, especially in the area of kefir on this. But we feel we're at the beginning of a new revolution, a new way of thinking about shaping brain during early life and critical windows by targeting the microbiome. And we have a number of success stories coming through in our animal models, and we're moving slowly into humans on this. And this is a very exciting area. So I'll leave it at that and thank a big group of people in my team here in Cork from all over the world. And I'll leave you with the other words of Hippocrates, who's also thought to have said, let food be thy medicine, and I'll change that to let food for your microbes be medicine for your mind. Thank you very much, and I'm happy to take questions.
Muchísimas gracias por la invitación a este evento. Para mí es un placer poder estar con todos ustedes.
Thank you for your invitation to this symposium. It's a great pleasure to speak to you all about the microbiome of breast milk and the influence in the development. We all know that maternal microbiota is one of the most important factors to determine, to impact the child health. So any early exposure during gestation and the breastfeeding life stage will have a direct impact on the development of the child, and in parallel will have an effect on the programming and development of the immune system and the metabolic activity. We cannot define what a healthy microbiota is, but we can know that children that were born through a vaginal delivery term and following the exclusive breastfeeding patterns as recommended by the World Health Organization are associated to a lower risk of developing diseases. Therefore, we consider this is the standard microbiota. This is a very complex process which starts at the beginning of our lives and evolves in composition, in quantity and diversity of the species from the very first life stages over the infancy, childhood, adolescence, so as to become more or less stable when we become adults at 15 to 18 years of age. So different factors such as the type of delivery, exposure to antibiotics, how and when complementary feeding was introduced, exclusive breastfeeding habits, etc. All those factors will play a decisive role and will impact on the good health of the infant. So over the first early life, in parallel to this process, we have the development and maturation of the immune system, cognitive development, motor development of the child. So any effect that can modulate or modify this colonization process, the colonization of the gut, will have an impact both on the immune system and the neurocognitive development, and therefore the health of the child. We know for sure that there are certain bacteria that include short-chain fatty acids, vitamins, and some compounds such as neurotransmitters, polyamines, and other metabolites that play a biological role on newborn babies. We know that we inherit bacteria from our mothers, so they are pioneers, they are the initial colonizers of the gut. Those bacteria play a very important role in regulating and modulating gut homeostasis. They also play an important role in the maturation of the immune system, modulation of the carbohydrate, lipids, and proteins. They help in cell proliferation and differentiation. They play an important role in blood coagulation, etc. So they all have a direct impact on the development of the individual. There are more and more evidence that suggests that any impairment, any alteration in the microbiota are associated to a higher risk of disease. So several problems such as allergies, inflammatory problems, obesity, overweight, celiac disease, autism, and diabetes, etc., are related to impairments or alterations in the composition and the diversity of human microbiota. So let's see what's the impact of the development of maternal microbiota on the development of the child. Some bacteria are transferred through in the moment of delivery and also during the lactation period. So we need an adequate milieu so as to create the proper imprint or trace in the newborn that will have a direct impact on his or her health. We enter into contact with bacteria in 3 times. First of all, during gestation. There is a lot of debate about this, so this is not today's topic. Secondly, second moment of contact, the delivery. When there is vaginal delivery, there is a great exposure to bacteria because the baby gets in contact with the bacteria present in the vagina. And thirdly, we will focus on the third moment, lactation, the breastfeeding stage. This bond, this created between mother and child, will have a direct impact on the development of the gut microbiota. Several studies indicated that both the mode of delivery and breastfeeding habits play an important role. This study, published in Science International Medicine some years ago, indicate that mode of delivery can be modulated by the breastfeeding habits. So if the child is breastfed, the difference tend to be less notable, less remarkable. So it's suggested that mother breastfeeding modulates the impact of C-sections and especially exposure to antibiotics. There is a lot of scientific evidence about the good effects of breastfeeding. They say that children that were fed with human milk are less susceptible to gastrointestinal and respiratory infections. Most of the evidences are focused on the gut, but there are other niches on the human body that are inhabited by bacteria. Our team of researchers worked on this area, and we managed to describe that the mode of delivery and the breastfeeding habits play a very important role in the colonization of the oral microbiota. So we observed that the mode of delivery, especially exposure to antibiotics, played a very important role on the oral microbiotic, and this mark was observed up to 6 months of life. And from that onwards, we couldn't distinguish any differences. And this could be partly explained by the introduction of complementary feeding, or maybe because the infant is attending nurseries, in contact with more children, etc., etc. Regarding breastfeeding habits, we saw that those children, those babies that were partially breastfed over the first 6 months of life presented a oral microbiota trajectory that was completely different to those children that didn't follow exclusive breastfeeding habits over the first months of life. We also studied the role played by breastfeeding in the microbiota of preterm babies. We saw the effect of breast milk, human milk, donated milk, and formula milk. We observed that the preterm microbiotas receiving donated milk, this profile is very similar to the profile of the child that receives milk from his mother, whereas there are more important differences with the formula milk. This microbiota was quite different, whereas donated and mother milk were similar but somehow different. And this could well be due to several reasons. First of all, donated milk is pasteurized. This treatment reduces or modifies the bioactive compound and macronutrient profile of the milk. And secondly, donated milk comes from mothers that had their children, they had term babies, not preterm. So the composition of the milk tends to be slightly different. Composition of micronutrients and bioactive compounds is different. So breastfeeding will play a very important role in the colonization of the gut. So which factors make breast milk so important, so interesting beyond nutritional aspects, because this affects health of the infant short and long term. It's not easy at all to work with samples of breast milk. This is a very complex with sample with bioactive complexes, different compounds that is influenced by the timing of feeding, and it changes in the composition from the colostrum. This is the first type of milk we receive when we are born. Then it transitions into mature milk. We also know that breast milk changes within the same day. The composition is not the same if it's taken in the morning or at night. In terms of cortisol, serotonin, melatonin, they change their profile over the days. Some components follow circadian rhythms. Composition of the milk changes according to the feeding times, according to the moment the child is breastfed, or according to what the mother eats. There are some studies that indicate compositions can change from one breast to another. Recent studies suggest that the milk changes depending on whether the child is a boy, a female, or a male. We are learning a lot. We know that breast milk is a model for immune tolerance because the milk transfers antigens and immunomodulating compounds, immunological compounds, hormones, and also HMOs, bacteria and metabolites. All these will regulate the immaturity we have in our immune system at birth, and this will foster the adequate milieu. So in short, breastfeeding, apart from satisfying from meeting nutritional requirements of the infant, it provides a great variety of immunoactive components, and I would like to stress the very important variety of bacteria that are transferred from breast milk. And with all these components, these are the first colonizers we receive from our mother at the moment of delivery, with direct effects, as we said, in the development of the immune system, protection against pathogen infections. It supports gut homeostasis, and it reduces the risk of having intestinal diseases. Breast milk is a constant source of microorganisms. We used to think that it was sterile up to 2003, but thanks to a very important work from Spain, we managed to demonstrate that the breast milk contained very important bacteria, such as lactobacillus and bifidobacterium. Thanks to the development of new technologies, for instance, massive sequencing, we've increased our knowledge, and around 800 different species of bacteria have been described mostly from Staphylococcus and Streptococcus, although we also find lactic acid, phylobacteria, and also protobacteria such as Escherichia coli and Klebsiella, and they are all in breast milk. So the quantity in absolute terms is very, very, very low. This is the colony forming units per milliliter. But if we think that the child receives around a liter of milk per day, this constant supply of my bacteria over a long period of time will play a decisive role. Which are the important functions? Structurally, because it fosters homeostasis, it reinforces the barrier function, metabolic also, because most of these bacteria are capable of producing vitamins, short-chain fatty acids. They help mature the immune system, they prevent pathogens or other toxic components coming from the diet, will be modulate somehow. So as I said, it plays a very important role. The presence of bacteria in breast milk is not exclusive of human beings. We found the presence of bacteria in the milk of other mammals. Interestingly, there is a great degree of variability across women and across times. As you can see in the slide, each bar represents a specific time, and each color represents a bacteria group. The profile S6 of one mother has nothing to do with profile H8 of another mother. If we try to compare microbiota of human milk with other milieu in the body, we see that the clustering is quite different from microbiota from the skin, from our mouth, of our vagina. So this suggests that there is a specific milieu, but we cannot forget that we can find other type of microorganisms in breast milk. Our team, the members of our teams, were pioneers in showing the presence of fungi in breast milk samples. This was accidental. We were studying, and we found out a colony, a very creamy sample, and looking at under the microscope, it was like a yeast, and by using specific techniques, we found out it was a type of yeast. So by using different cultures and different methods, among others, massive sequences, we could confirm the presence of yeasts, fungi, etc. For instance, Saccharomyces cerevisiae, a very common yeast in foodstuffs, was present in the breast milk. So feeding habits and the environment could be one of the main important reasons of the presence of yeast and fungi. Although we can also find viruses, archaeas, and phages. So the complexity and the variety of microorganisms in the human milk is really impressive. So we still don't know what's the biological role they play in the colonization and the modulation of the immune system of the child, and therefore in its health. Apart from the complexity, we know that the same perinatal factors that affect the child microbiota affect maternal microbiota. And this has a reflection on the milk, on the type of milk. For instance, medicine consumption, maternal diet, of course, the lactation on the stage, environment, maternal health, etc., etc. And if we add the high complexity of the breast milk composition, apart from bacteria and other microorganisms, we find other bioactive components such as cytokines, growth factors, HMOs, hormones, proteins, and peptides, fatty acids. As a result, we can just start to glimpse the colossal complexity of breast milk. We need to understand which are the specific factors modulating this composition, which is the interrelation between the different components, and which are the effects on the newborn health in terms of growth, development of microbiota, reduction of infection risks, allergies, asthma development, etc., etc. Therefore, we can say that there is a direct relationship between feeding habits, microbiome, and immune system, and this is related with the risk of illness. So we need to understand the role played by diet and the transfer of bacteria and the role they play in the child biota, because this will help us develop new dietary strategies geared at programming that will support child development and will support nutritional, microbiological, immunological programming over the first 1,000 years of life. And I would like to finish my presentation by thanking my team and all the funding sources that made possible this work in this field of research, microbiota and lactation. I will be happy to answer to any of your questions. Thank you.
Hello to everybody. I'm happy to participate in this symposium. And my name is Antonio Suarez. I work at the University of Granada in Spain. And I want to show you some data that we have collected from the cognitive study, specifically on how the enrichment with milk fat global membranes and LC-PUFA and symbiotics have changed the pattern of microbiome maturation and evolution in children. In the talk, I will show you how the microbiota has changed in the cognitive study from the first month to the 18 months of life. And we did this experiment first by classifying the microbiota by enterotypes. Then we studied the enterotypes and studied their structure, the composition and diversity, to move to the evolution. We have 4 time points for each child, and we wanted to know whether the enterotypes changed during the evolution of the infants. And we tested it in connection with the time points, with the age and the intervention of the study. We studied also the dynamics. The dynamics, I will show you data on how, case by case, the children evolved, changing their enterotypes. And that's what we call trajectories. And I will show you that the children have different trajectories on how their enterotypes evolve, depending on the diet, the intervention study. And finally, I will show you the last data on the impact of the new formula on the probiotics in children. Well, we all know that the breast milk composition is the gold standard. It's composed by many different compounds. And I want to stress that the experimental formula of the cognitive study was enriched in 2 lipid molecules, essentially in milk fat global membranes. It was enriched in this special sort of fat presentation. Also in LC-PUFA and in symbiotics. Symbiotics is a mixture between the addition of both the probiotic, the sugar, in this case, and the prebiotic, the bacteria. So the idea of the symbiotics is that you provide the sugar and the bacteria, so the sugar that promote the growth of the beneficial bacteria. So this formula, the difference with the previous formula is that it has been enriched in these three different compounds, with the idea of trying to mimic as much as possible the composition of human breast milk. The cognitive study was designed in the collection of samples in a long period. The collection has now ended the 6th year of sample and data retrieval from the infants. And there were 3 groups. The standard formula with 85 children, the new formula with the addition of these global membranes and the symbiotics and the LC-PUFA, and a control group of exclusively breastfed infants. And samples were collected in all these periods, and we will concentrate on the initial part of the evolution of the colonization of the gut by bacteria. So the first data I want to show you is the microbial perspective of the samples. Here you have the phylogeny. This means the classification, taxonomic classification of bacteria. On the right, you have the genera. We have retrieved the 30 most abundant genera in all the samples. Here we have more than 400 samples that were collected from the children. And you can see there are quite different taxonomical profiles from the first to the 18th month. So we had the initial data on the microbiota of these children. And we wanted to know whether the composition of this study was affected or was influenced by study variables. We have in the colony study a lot of different variables were collected from the parents and the children. And in this graph, what I show you is that are the collection of variables that had some significant effect on the microbiota composition. On the left, you have all So the age by far was the variable that most affected microbiota composition by far, followed by the type of feeding. Here we have the standard formula, experimental formula on the breastfed children, breastfeeding days, daycare, whether the children were in-home raised or in nursery or in school, the smoking, father smoking habits, antibiotics, diarrhea, and then some information on solid food intake, meat, fish, and fruits and vegetables. In the whole experiment, the variable with the highest impact was age, but when we stratify by months, we can then clearly see that type of feeding was extremely important at the 6, 12, and 18 months. Also with the breastfeeding, how many days was that child breastfed in the mixed feeding, how many days the child was breastfed. So these are by far the 3 most important variables in the study affecting microbiota composition. And then, as I said before, we searched for enterotypes. Enterotypes is a way of classification humans depending on the composition of the gut microbiota, of the overall composition of the gut microbiota. And this has been extensively studied and used in many different papers. And we wanted to test whether we could detect different configurations in the microbiota of the children in these 400 samples. So and we wanted to know whether the dietary intervention could affect the presentation of these enterotypes and their evolution, changing and shifts along the growing of the child. For that, we used a system. It's called the Dirichlet multinomial mixture components. It is a statistical system that allows for detection of enterotypes in microbiota samples. When we use this algorithm, the algorithm identified 4 different enterotypes, the least number of enterotypes that best describe the 400 samples. So it was quite surprising because we have in literature the number of enterotypes in this range of ages normally is 2 to 3 different enterotypes. And in our data set, the system identified 4 different enterotypes. These 4 different enterotypes are this is the heat map. Each line represents a genera that you see the name on the right side, and every vertical line is a sample. So the algorithm produced this heat map and told us that there were 4 different enterotypes, 2 reasonably big in the number of samples, and 2 with at least a reduced number of samples. They were depicted in gray and red. So we had the 4 enterotypes, and we had the genera that drive these enterotypes. And these are the genera that drive the configuration of the different enterotypes. And on the left side, we have the drivers of the first enterotype. These are the bacteria that determine this enterotype. And if you see on the left, there are different colors because some of them belong to the Firmicutes taxa in orange. Some belong to Bacteroidetes in blue, and some of them are Proteobacteria. Indeed, when you look at the taxonomical names, it is a mix of aerobic and anaerobic bacteria. So when we determined this enterotype, we decided to call it mixed, the mixed enterotype, mixed anaerobic, mixed aerobic bacteria. The second enterotype was determined mostly by the abundance of one genera, and that genera is the unclassified Drangospiraceae. So since this enterotype was determined by this genera, we called it ULAC, from unclassified Drangospiraceae. The third enterotype is also dominated by a single genera, though some others contribute to the profiling of this enterotype, but it's mostly determined by the abundance of bacteroides in this case. So we call it BACT. And the last one was determined mostly by two genera, but all of them, except for bacteroides, they all belong to the Firmicutes phylum. So in this case, we decided to call it FIRM. So we have 4 enterotypes. The first one with this mix of aerobic and anaerobic bacteria, two determined by two genera and classified among Spiraceae and bacteroides, and the last one by a mix of Firmicutes bacteria. When we looked at the data, we in this algorithm, we introduced 87 genera, 87. Only 19 were enough to describe this enterotype. So these are the most important genera in our samples to define the enterotypes, only 19. The contribution to the clustering of these enterotypes, to the organization, is quite dissimilar. For example, bacteroides is present as a driver in the 4 enterotypes, but mostly in the 3rd, as we showed previously. Unclassified Spiraceae is also present in the 4 different enterotypes, but determines more the enterotype in the EULAC and FIRM, and then the rest of the genera are present in some of the other enterotypes. In a few, the last ones are exclusively present in one enterotype, like for example, Enterococcus, Klebsiella, Lactobacillus, Clostridium, and Escherichia. They are present exclusively in the mixed enterotype. So these 19 genera really define the most important drivers of the different enterotypes. And we wondered, these 19 genera, how much do they account for the overall composition of the microbiota? And as you can see here, these genera accounted for more than 85% of all the readings in the microbiota experiment. So these are obviously the most important genera in abundance in our sample set, and they cover more than 70, depending the firms, the one with the least accounting data, but it's over 75, more than almost 80% of the microbiota. The composition of the enterotypes is quite different. So on the left, you have the Firmicutes bacteroides ratio that has been extensively used in microbiota studies. And of course, the one with the lowest ratio is the BACT enterotype. And on your right, you have the composition at phyla level, and where you have actinobacteria in blue, bacteroides in green, Firmicutes in red, and Protobacteria in purple. And you can see that there are 4 very different distributions of composition in the 4 different sets of enterotypes. So they are different in composition, and they are also in diversity. This is quite important because for a long time, there's still a debate whether the alpha diversity of bacteria do truly inform about the health of sample. Still, we use these alpha diversity indexes on top the number of species that increases over with the enterotypes, the least number of species in the mixed enterotype, and the highest in the last one, the FIRM. And when we look at the diversity in the middle, also diversity increases with the different enterotypes. And finally, the last one on the bottom, the phylogenetic diversity. So they not only have more species with increased diversity, but they are also phylogenetically different. So this corresponds with the data that we already had, and then we studied the beta structure of the samples. We used the Bragg-Curtis distance to in a double space try to define whether the samples were similar or dissimilar between themselves. And the data show that those belonging to the mixed enterotype were quite dissimilar to the rest of the sample. And if you look at the dots, the quadrangle corresponds to the first month and the cross to the 6th month. So apparently, this was the first data we had that the mixed enterotype was present in the 1st and 6th month of the samples. Then we had the ULAC enterotype that was quite dissimilar to the mixed one. And then we had the BACT also in a separated group and the firm enterotype. And this separation was significant, showing that the composition of the 4 enterotypes were fairly different between the data. But then, do enterotypes associate with the most important variables in our study? The most important variables in our study were, I remind you, the time, the chronosequence of the cognitive study, 1 month, 6 month, 12 month, 18 month, and the type of feeding, because this is an intervention study. So we made this test. It is a statistical test where we make 3 associations. We associate the enterotypes on the left with the time of the sampling time on the top, and with the feeding group on the right. From this plot, we could significantly determine that the first month of age in our samples, most samples belong to the mixed enterotype. The sixth month had a mix of sample that had the mixed enterotype or the ULAC enterotype. The 12th month set of samples was more enriched in the ULAC and BACT enterotype, and finally the last 18-month samples were enriched in ULAC, BACT, and most of it in the firm enterotype. So, as you can see down, mixed ULAC, ULAC, BACT, apparently there is a significant association. So that there is an evolution of the enterotypes. Apparently, mixed is more enterotype of the beginning of the colonization, while the firm enterotype is more the last part. BACT and firm enterotype are more of the last part of the colonization in the children. So this is depending on age. What happens when we look at the data depending on the diet? Well, the breastfed infants only show in these 4 time points, they only show the mixed and ULAC enterotypes. This was surprising because we thought that there would be a mix of enterotypes in these children, but they only showed the mix and the ULAC enterotype. When you looked at the standard formula, apparently the children start all with the mixed enterotype, but then the transition. At the 6th, they are more in the ULAC, and in the 12th month, they are more in the ULAC, in the BACT enterotype, and finally, in the 18th month, they have more samples in the firm enterotype. So there is a time evolution associated with the diet. And when we looked at the experimental data, we found a different evolution. Apparently, at the 6th month, samples were a mix between the mixed enterotype on top, the ULAC enterotype in the middle, and the firm enterotype at the bottom. So there was some sort of effect from the experimental formula on the enterotype evolution in children compared with the standard formula. Significant. So we used this model, the Markov chain model, to understand how these enterotypes were transitioning with the time in the children. And as an example, I show first the breastfed enterotypes. On the blue is the mixed. I told you almost all children belong to the mixed and ULAC. You can see that the mixed enterotype, half of the samples still retain this enterotype with time, and the other half of the samples evolved to the ULAC enterotype in yellow. And the samples, when the children are in the ULAC enterotype, 83% of the samples remain in this enterotype. And just one move to 17%, and 7% of the samples move to the firm or backed enterotype. So apparently, breastfed infants, exclusively breastfed infants, stay in the mixed and ULAC enterotypes. When we looked at the standard formula, the dark shadow lines showed you the most important changes in the transitions of the enterotypes. Apparently, they all started in the mix in blue on top, but most of them, 55%, moved to the ULAC enterotype. At this time point, a few, 23, stayed in this enterotype, but 50% again moved to the back enterotype in gray on the bottom. At this stage, 50% again stayed at this enterotype, but then 30% moved to the firm enterotype. In red, what I show you is backward transitions when the sample is in the back enterotype, and then the last sample returns to the ULAC enterotype. In this case, the 18%. So that in this evolution, there are some children where the enterotypes go backwards in time to the previous most abundant enterotype. In the experimental formula, the transitions were fairly different from the standard formula, as I showed you before. And they started in the mix. The 41% goes to the ULAC enterotype, but then at this stage, 42% of the samples move to the firm enterotype, and those that go to the back enterotype at the bottom, most of them go to the firm. But apparently, the firm enterotype is more the last stage of these children. There are also transitions in these samples, retrogressions, where the enterotypes go backwards. So there are children that reach the firm enterotype, but 50% of them return to a previous enterotype, in this case, the back enterotype. So the enterotypes in the different sets of children were evolving in different manners. When we separated the cases by children and we searched for the evolution of the enterotypes, we detected 26 different ways of evolving in time. Firm in the 18 months, there are cases where the children go to a previous enterotype. We looked at these special cases where there was these changes in the evolution of the enterotypes, and we identified that there was a significant association with the diarrhea events. So apparently, diarrhea is a major disturbance of the trajectories, and when children suffer diarrhea, there is a high chance that the configuration of the whole microbiota changes and goes back to a previous stage. The prevalence, how many children show these trajectories? Well, this is the prevalence of the children of the samples. And as you can see, breastfed, in this case in blue, they are at the bottom of the trajectories. And red samples are the standard formula. They are mostly in the upper part of the trajectories, and apparently the experimental formula is distributed along the 26 trajectories. When we obtained this graph, we thought that there might be some sort of shift of depending on the type of feeding. Like type of feeding was affecting the trajectories of the children, the preference of trajectories. And when we looked at the density distribution, we could see this effect more clearly in the deleting the retrogressions, those that go back. Okay, when you delete the retrogressions, you could see that the density, the samples were not distributed homogeneously in their trajectories. The blue, in this case, the breastfed infants, as you can see, they mostly belong to the trajectories on the left, those with a high number of mixed and fertile. The red sample, this belongs to the standard formula, so there is a slope that goes steadily up to the right of the density graph, indicating that most children had used this kind of trajectories. But when we looked at the experimental formula, there are two hills. Look like half of the samples used trajectories similarly used by breastfed, and half of the samples were using trajectories similarly to the standard formula. We used a statistical test to determine whether this association was right, and that was true because the Kolmogorov-Smirnov test told us where there was the slopes and the form of the modality of this density plot was similar between the sample, and the only difference was observed between the standard formula and the breastfed, because the breastfed, the blue one, and then the standard formula goes to the right. And when we looked at the modality test, the Hartigan's D test told us that the standard formula was a bimodal formula, or a bimodal graph. So it was true that there are two sets of samples in the standard formula. We separated these samples depending on their trajectories, and we stratified these children in two different sets. The one on the right that we called the fast trajectory, because as you can see, the trajectories goes from the mix in blue, immediately change to ULAC or BAC. They immediately change at the second stage, the 6th month. And the slow children at the 6th month, they still had the mixed enterotype. There were even children that at the 12th month still had the mixed enterotype, like the breastfed children. So the experimental formula had produced two different trajectories. One fast, like the standard formula before, enterotypes change in a timely manner, very fast, and slow children. And in these children, the enterotypes were evolving more slowly. They still kept the mixed enterotype, and they required 1 year to detect that there was a change in the enterotypes in some of them. So we stratified them in fast and slow. Why do we have two different trajectories? The fast and the slow, that is very similar to breastfed infants. So in this case, what we did was logistic regression analysis, and we could see that 3 variables were determining the evolution of the children: the maternal BMI, the delivery mode, and the daycare assistance of the children. And we could obtain from this formula that the slow infants, those whose trajectory is mostly similar to breastfed, are experimental formula fed infants that have been in-home care they have been born by vaginal delivery and they have been born to lean mothers. So diet is a major influence. The experimental formula produces a microbiota evolution very similar to the breastfed when these three conditions are also met by the mother and the lifestyle of the child. This is a major result because it's the first time that the experimental formula shows a maturation that develops as the breastfed infant when these three conditions are also met by the family and the lifestyle of the infant. The last thing we did was when we studied the compositional differences between the formulas, and we focused on the probiotics. And there were significant differences between the standard formula and the experimental formula. But focusing on probiotics, we could see that the experimental formula on your right, and the bigger dot means that the data is significant. It produced a significant fold change in the growth of all these taxa, and specifically of lactobacillus that was significantly enriched in the 6 and the 12 and the 18-month samples, and bifidobacterium only in the 18 months of the children that were fed the experimental formula, while the standard formula only influenced significantly onto Eotius. So in conclusion, what I've shown you in this experiment is that we have identified 4 enterotypes. One of them has never been previously described in literature, the EULAC enterotype. The enterotypes substantially differ in the composition and structure. Transitions were strongly associated with age and the type of feeding. So enterotypes depend on these 2 variables. We detected 26 different ways of evolutions. A few of them were backwards retrogressions that were determined by diarrhea events in the children. The essential result of this experiment is that experimental formula-fed infants evolved like breastfed infants when lean mother, in-home care, and vaginal delivery also are covariables in the children. And the last is that the experimental formula, as I showed you, and has a lactobacillus and bifidobacterium growth in the children. I hope that you enjoy this talk, and I'm waiting, and I'm looking forward for your questions on our study. Thank you very much.
Okay. Thank you very much, the 3 presenters. Excellent talks. And we have several questions now to be answered. There are only a few, so I think we will be able to answer everything. So the first one, it is a question saying that what is the dose of probiotics for neonates and infants with gastrointestinal diseases, and what forms? Drops, sackets, beds, components? So perhaps John can answer about that if there are any data. Or Carmen.
Yeah, it was probably more for Carmen than for me. Sorry, Carmen.
Well, I think first of all, thank you very much for the invitation. For me, it has been a pleasure to be here. Second, regarding to the question, I think we need to identify which is the scientific evidence, because at the end we need to recommend the things according to the evidence. Then we need to test the specific strains, the specific dose, a specific matrix for specific diseases in kids. Usually the probiotics are provided to the kids in drops, but also in lyophilized form within the formulas or within other type of products. But of course, regarding to the recommendations, always I would like to suggest to take a look to the literature and to check the evidence and recommend just using the evidence base and with the specific strains and specific products. That's my point and the most important thing.
Right.
Okay, thank you, Dr. Carmen Collado. And now we continue with the next question. Is there a blood or a stool test to assess sufficiency of probiotics in the gut of infants? Do you know that? Dr. Collado, perhaps also?
Well, no, I don't know if the quantity of bifido, but I know that there is a test in US that they are trying to study the quantity of acetate, because acetate is one of the main metabolites produced by bacteria. Then if they by bifido, sorry, then if they identify that there is a lower amounts of acetate in fecal supernatant, then they suggest to be under to consume probiotics, mainly bifido. But I don't know in Spain or in Europe, what is the situation? I have no idea.
Professor Antonio, do you know something about that? To add something?
No, I've never heard, because it's very difficult to,
Okay.
I think it's very difficult to define a healthy enterotype.
Si me dejáis, es que
Sí,
puedo,
yes.
sí.
Yes.
Can I add something?
Hay
Yep.
compañías,
There are some companies
en Europa y en Estados
in
Unidos
Europe and in
que
the
te hacen el
United States
perfil
that provide
de
a profile,
microbiota
a microbiota
en las
profile
heces, y dentro de ese
based
perfil de
on
microbiota
stools,
puedes
and
encontrar la
within that
presencia de
profile you can
lactobacillus o
find the
bifidobacterias,
presence of
que suelen
lactobacillus or
ser los probióticos más
bifidobacteria, which
habituales
are the most
utilizados
common
en la industria
probiotics
y en la alimentación infantil.
used both in child foodstuffs and in the industry.
Next question. Said, dear professor, thank you for the nice and clear presentation. And I'd like to know how external environmental milieu could shape our microbiota's diversity and the impact for human life. Perhaps John?
Yeah, so I mean, this is a really exciting area of trying to understand about the relationship between the environment we live in and the microbiome, and particularly in early life. A lot of work now in cohort studies is trying to really dissect out what are the different influences. And so some of the key things that people are beginning to see is living in a green area, green space with exposure to lots of outdoor aspects is affecting the microbiome. Everything from that to having a pet, whether you're close to animals. These are also things that will influence the microbiome early in life. There's some really good work out of Susan Lynch's lab in the US, looking at various factors, that will influence what's going on. So, I mean, to be honest, the microbiome is really like a conduit for kind of mirroring what's going on in the external environment and how it can actually influence what's going on. So it really is a way of how the environment can shape our internal physiology.
Thank you very much. Next question said Samazar, we know that bacterial alpha diversity and abundance difference between premature and term babies, especially during the first 6 weeks after delivery. And so how can increase the number and efficacy of microbiota for premature babies, especially during the first 6 weeks after delivery? This question for Carmen, perhaps?
Or maybe to Antonio.
Or
I don't
Antonio.
know. These 6 weeks is quite tight. I think it's so complicated to answer this question, Antonio.
Yeah.
What do you think?
I talked to one pediatrician in a hospital, and they were trying to provide the fecal bacteria from the mother, and there was an idea, and they thought that it would be useful. So I think they were trying to in some way also from the breast milk, they were trying to give the children the bacteria from breast milk and feces. So they were working on that idea. They were not sure whether it would be good, but they were working on that idea, because it's a way of you colonize the children in the infants in the way they should have been colonized. And that might be some line to explore, because I think it would be perhaps the easiest way.
Yeah, I agree that in the premature NICUs, we need to promote the breastfeeding practices as much as we can, and also to promote the donor milk, because despite the donor milk is suffering some heat treatment, at the end it contains different bioactive compounds that can also promote an adequate microbial colonization and an adequate immune stimulation. Other environmental sources would be the skin to skin with the mothers, the kangaroo method, and all these things that at the end they are quite relevant for this microbial environment. Then I think to try to promote an adequate environment in preterms is breastfeeding and also skin to skin and well, family environment.
Yes, I agree with all your comments. I think that the studies are ongoing in preterm infants, and the human milk is, by the moment, the first source for research, and also to improve the probiotics supply for the preterm. And there are several other questions. Here, another question saying, can preterm milk products increase the weight for more than 3 months babies? Okay, I can say here that these products, the preterm milk products, are already prepared. It's a formula prepared for preterm babies. I do not know if there are some studies showing this increase of weight in preterm babies younger than 3 months. You know that about that, Jose Antonio? But exactly the weight.
And I don't know exactly if there is clinical trials especially for the increase of the weight, but in my opinion, the pediatrician follow the weight of the babies, and has to be the pediatrician that the person that have to decide if the baby has to change the preterm formula to an initial formula, do the pass, but the products that we have in the preterm milk products can increase the weight for the babies. Yeah, but has to be on the supervision of the pediatrician, of course.
Mm-hmm. There are also another question saying that if there are any specific time to indicate probiotics, and how many times should be administered. By the moment, from my point of view, there are some infant formulas who contain probiotics used to be have to be assessed already by different studies, and there are only a few of these probiotics already approved by the authorities. So the question is, it's not a specific time. Of course, as early as possible, the effect will be stronger. And if you maintain on the time, the question is that it should be the correct probiotic. And most of the time is given with prebiotics at the same time as symbiotics. The question is that there are still the need for further studies to understand very well how many times is enough, and also if there are a mix of probiotics, the perfect one to promote the best, the optimal development of the gut microbiota. But from my perspective, to be honest, as soon as you give this probiotic, the effects will be stronger. The problem is if you have the right one. We need to do further studies. As you can see in the study of who has presented, the Colonies study, who has presented Antonio, as you see, we provided some probiotics with the infant formula, and the effects has long-term effects, and also closer to the to babies breastfed, but not still the same that the breastfed infants. We need to do further research. I don't know if John want to add something, or Carmen, or Antonio.
No, I totally agree, Christina. I think, we just don't know enough. We have to also be very careful, as you say, the word probiotic is used a lot, and we lump things together, and we have to really build in a more precision into understanding about what strain of bacteria at what dose at what time for how long, and we're just not there yet. We need a lot more data, but it is very encouraging, and I agree with you, the earlier the better.
Yeah, I also would like to point it out that probiotics and prebiotics, not as a supplements, but within the diet, have been consumed during all our history. We evolved with fermented milks, we evolved with fibers and all these things that at the end they are exerting a specific effect in our gut. Of course, in specific periods in our time, we need an extra of these products, and then we can use these supplements, probiotics and prebiotics. But I think we need also to take care of the maternal microbiota and maternal diet during the pregnancy or even before, and also during the lactation, because at the end there is a transfer from moms to infants, and we need to start to thinking on how to take care of our microbes as well.
Okay, there are a few questions, but we are out of time. I'm so sorry. We need to move forward, otherwise we will finish very late today. So I will share the questions with the presenters, speakers, because there are not too many. And if they want to answer you, you have also, I will share with you and you can answer them. Thank you very much again for your disposition, always very nice to saying yes always, and I'm really glad to have you on board in this session. Thank you very much. Very well done.
Thank you.
Everybody.
Thank you.
Thank you. Thank you.
Bye.
Bye.
Buenas tardes, pues agradeciendo la participación de toda la
Good
mesa.
evening. I would like to thank the prior panel and their scientific value and clarity. And now we have the next panel regarding the diet of infants and its impact on their immunological development system. And for this panel, we have Dr. Cristina Campoy. I think everybody knows her. She has participated in this Ordesa chair very actively, and it's wonderful to be able to count on her. She's a full university professor at the University of Granada, and she is also responsible for the investigation group of metabolism in pediatrics. She's the director of the Cátedra Ordesa at the University of Granada. She's coordinator of the official inter-university master on genetic, nutrition and environmental factors, NutriVigen, and GD factors, and she's a member of different scientific groups such as ESCAN, and she has written many different papers in national and international journals. And with us in this second panel, we also have a very well-known panelist, Dr. Jose Manuel Moreno Villares, who studied medicine at the Complutense University of Madrid, and he has worked also at the California University. He has worked at the University Hospital 12 de Octubre in Madrid, and he currently works in the pediatrics department of the Hospital University of Navarra, and he is co-director of the pediatrics department at the Navarra University and a collaborating professor. And lastly, we have Dr. Ascensión Marcos, who will introduce us to Dr. Philip Calder. Thank you very much, Maria Jesus. Professor Philip Calder is head of the School of Human Development and Health and professor of nutritional immunology in the Faculty of Medicine of the University of Southampton in the UK. He is an international recognized researcher on the metabolism and functionality of fatty acids, with an emphasis on the roles of omega-3 fatty acids, looking at the influence of diet and nutrients on the immune and inflammatory responses. He's received many awards and prizes for his work, from Danone, for example, in 2016, and he's also the president of the International Society for the Study of Fatty Acids and Lipids, and he's also been the chair of the Scientific Committee of the European Society for Clinical Nutrition and Metabolism, ESPEN, and he has also been president of the Nutrition Society, the British Nutrition Society. He is currently president of the Federation of European Nutrition Societies, and he is co-author of several reports of the ESPEN expert group, and also he has been the editor-in-chief of several journals, such as the British Journal of Nutrition and the Journal of Nutrition, Clinical Science, Nutrition Research, and Annals of Nutrition and Metabolism. And I am convinced that with these speakers, as in the previous session, we're going to have a wonderful panel. It's going to be wonderful to listen to them. So the floor is yours.
Buenos días a todos. Es para mí un honor.
Good evening. It's an honor for me to be part of this conference, this 7th International Scientific Digital Symposium. Well, in this case, it's digital. And I am very happy to be here with you today and to talk about the early programming of the immune system is a challenge. And we already heard in the previous session how Dr. Collada and Dr. Ryan have spoken about this topic, and we're going to insist on this topic because we believe that it's very interesting today, because the immune system starts developing throughout gestation and pregnancy with an adaptive immunity. You see on this chart how it starts in the first term of pregnancy with some dependent T cells and B cell dependent. And in the second and third term of pregnancy, we start seeing a release of some interleukins that are associated with innate immunity. But until birth, what we mainly see is a transfer of the IgG maternal antibodies, and the child will also receive the IgA antibodies through breast milk, and the IgG antibodies that go through the placenta to the fetus in the last part of gestation have a very important role in the tolerant response to food and other allergens, and with regard to the immune response of the infant. Additionally, after birth, we see the T cells and the natural killer T cells and the T regulatory cells, and we see higher levels of interleukin 10 and 12 and also interferon alpha. And in the case of interleukin 12, we can see how it starts at a low level and then it later increases. However, the development and maturity of the immune system doesn't end until adulthood in many cases. And it's important to bear in mind that we have this early programming and that there are some factors that are very important for the development of the immune system. And we also know that the development of the immune system is very much associated with some chronic diseases with that are inflammatory autoimmune diseases. So there are different factors. For example, maternal malnutrition, obesity, or diabetes, or the activation of the maternal HPA axis, or an impairment of the maternal immunocompetence, and there is a lower maternal IgG placental transfer, or a reduction of human immunological factors. All this can have an effect, an impact on the fetal development and on the development of the immune cells that are learning, so to speak, to provide certain responses after birth. All this is associated with the development of the infant at a metabolic level, and this is associated also with chronic inflammatory diseases such as the metabolic syndrome, cardiovascular diseases, cancer, or diabetes, or autoimmune diseases. And today we know that there are also epigenetic factors that will have an impact on the development of immune cells. If we take into account all these factors that are involved in the development of the immune system and the development of allergies, we can see the fact that they are factors that have to do with the microbiota, such as the type of delivery, the genetic predisposition, the use of antibiotics during pregnancy, unhealthy lifestyle, environmental contaminants, the type of diet, the status of the maternal microbiota and how it's transferred to the fetus, and the fact of receiving feeding breast milk or formula. So we have these epigenetic factors that can have an impact on the programming of the immune cells, and this will lead during gestation to a transient hematopoiesis of these cells, and this will determine how they are going to behave and their function in the case of macrophages and mast cells. And all this will impact, depending on this imprinting, will be determining the presence of long-term diseases. This slide is particularly interesting because it assesses how nutritional parameters are very important during the first stages of life. We see how maternal nutrition plays a very important role in the increase of risk of having diseases that are associated with the immune system or chronic diseases that are inflammatory. And depending on the mother's diet, if we have a mother that takes an appropriate number of calories, then there'll be a better fetal development. And if the mother also takes N3 PUFAs, this will reduce the risk of atopia. And if the mother has got dysbiosis, this will have an effect on the immunity. It will lower the antibodies. that are transferred to the offspring. And if we have maternal malnutrition, there'll be a reduction in the child of the immune response to vaccines. And if we have malnutrition in mothers with zinc deficit, then there's an increase of cortisol levels, and this can have an impact on the patient's erythropoiesis. Additionally, when the child is born, it's important to see the difference between breastfeeding and formula, because breastfed infants will have an important intake of nutrients and will also receive bacteria, microbiome from the mother that will lead to an improvement of the innate immune system, and they will also be receiving oligosaccharides that will protect the gut mucosa that's under development, and also the immunoglobulins. And we shouldn't forget that the immune system continues to develop until 2 years of age, and therefore nutrition has a key role, and we reduce the risk of allergies and inflammatory diseases if there is a nutrition rich in fruit and vegetables. And children who have a deficit of iron, vitamin B12, vitamin D, and zinc will see a negative impact on their immune system. And also in children with a reduced protein intake, this can lead to an atrophy of primary lymphoid organs, and this can alter their immune development and increase the risk of diseases in the long term. Therefore, nutrition has a wide impact, and for example, in the case of allergies, we see that a high intake of vitamins, minerals through fruit and vegetables and will have an effect in the same way as the intake of prebiotics and probiotics or fiber that can be protective, and the same happens with N3 PUFAs. And therefore, we can say that it's possible to design a diet, or we can intervene with a diet to prevent diseases during this critical period from the prenatal phase until 2 years of age. So in this way, we can stop these diseases from occurring, from developing. And why is that? Because we have to take into account the timing of the GI tract that is very long in humans and doesn't end until childhood, until 6 or 7 years of age or even later. Therefore, the most sensitive stage would be the prenatal stage and the postnatal stage until 2 years of age, where there is a development of the epithelial cells and of the gut mucosa. Additionally, during this time, there's an implementation of the microbiota. It's the initial touch point with foodstuffs, of the body with foodstuffs, and the reactions in the GI tract will have an impact on the development of the immune system. I like to highlight the fact that the shaping of the immune system starts with the maternal microbiota, and during pregnancy and lactation, there is an imprinting of the gut microbiota that will determine the reaction of the child when faced with different stimuli, different allergens, and this will influence the infant's development depending on the child's nutrition, on genetic factors, the health of the mother, environmental factors, stress, infections, medication, and so on, and the type of diet and the use of prebiotics during these very critical stages of development. I have to say that when we are talking about the early programming of immune system, I have to say that breastfeeding provides the best nutrition to newborns, to infants, and compared with formula-fed infants, the growth patterns are different. We also see different nutritional status, a different gut microflora, and fewer infections in breastfed infants versus formula-fed infants. And there's also a different immune system development and immune response and a better neurodevelopment, and there's an innate programming of the taste that the infants will prefer, and a prolonged exclusive breastfeeding will reduce the risk of GI respiratory infections and hospitalization for infections, even in developed countries. Therefore, breastfeeding has many important advantages. Firstly, we have the transference of multiple immune factors, and it promotes the gut immunity, and it also has an anti-inflammatory effect. Everything indicates, therefore, that breast milk is the perfect nutrition for the development of the immune system. In fact, breastfeeding transfers in a passive way IgA antibodies. It also regulates the development and the function of the gut permeability. Furthermore, it offers bioactive antimicrobial factors to the child, and it has a modulating and an immunoregulating effect on the evolution of the immune system, and it offers anti-inflammatory and antimicrobial agents that are very important during the early stages of life. And breastfeeding includes some very important ingredients such as proteins and lipids and carbohydrates and vitamins and minerals. And additionally, it offers bioactive components such as B and T lymphocytes, macrophages, immunoglobulins, growth factors, FGM components, the LC, PUFAs, the HMOs, probiotics, prebiotics, and other bioactive peptides that will have a very important impact on the development of the infant's immune system. And human breast milk also includes non-nutritive biomolecules such as bacteria, archaea, viruses, protozoa, anaerobic fungi, and lactic acid bacteria that are important and very well known due to their beneficial roles, such as Lactobacillus casei, Lactobacillus fermentum, and also the Bifidobacterium and the Bacteroides and the Clostridia class that will also be implanted in the gut and that are very important for this immune system development. We are talking about the core human breast milk bacterium that has Lactobacillus, Staphylococcus, Streptococcus, and Proteobacterium. All these bacteria of the bacterium may seem pathogenic, but when received through breast milk, then these bacteria will have a very important role in a positive development of the infant. And this microbial pattern in the gut, as I say, is very important. So breast milk has an important immune modulation effect that will sustain and support and regulate the cell population in the immune system, and will also help with the maturation of the infant gut and associated lymphoid tissues, and also through the cytokines and the chemokines and the PUFAs, the MFGM, the nucleotides and glycoproteins and oligosaccharides. And all of this will be associated with the development of diseases such as allergy or asthma. So breast milk versus infant formula will create an immune tolerance network at the gut that is based on the interaction between the microbiome and the gut immune system, and this favors the optimal development of the infant's immune system. And I'd like to highlight the HMOs as very important components. Some mothers secrete HMOs, some others don't. And HMOs are in breast milk, and they're associated with the microbiota of the mother. And this differs depending on whether the mother secretes HMOs or not. And the HMOs will drive the infant gut microbial colonization, and the maternal secretor status will also have an impact on the neonatal gut microbiota. It's not the same to have a mother who secretes HMOs versus one that doesn't. And when there's a disruption of the early microbiota colonization, this has been associated with the risk of disease. Therefore, it's important to have this HMO secretion and to have an optimal microbiome in breast milk. Another important component that also impacts the development of the immune system is the MFGM. And there are several studies that confirm its effect on the development of the brain, and there's very important data regarding the benefits for the immune system development and the gut, and we see a lower prevalence of diarrhea in infants that are receiving formula supplemented with these components, and there's a reduction of otitis media, and there's also a bactericidal activity against several foodborne pathogens and a lower incidence of gastrointestinal and respiratory infections. Additionally, we see a benefit on the development of the intestinal epithelium. There's a protection against inflammation induced by Clostridium difficile toxin, and we observe a better microbiota profile that's more similar to that of subjects receiving breast milk, and it also has a moderate effect on the shaping of the gut microbial activity and function. When the MFGM and probiotics are added to the nutrition, this will have a significant impact on the composition of the microbiota. Lastly, I would like to make a reference to complementary supplementary nutrition and its impact on allergies, because it's maybe possible to have some primary prevention with an early exposure to food allergens to induce tolerance and reduce allergies. And it has been shown that the timing of allergenic food introduction is not associated with other allergic outcomes. And although the findings cannot be generalized beyond food allergy, we see that there is this mediation when there's IgE antibodies. And I wanted to mention this slide because the immune system continues to develop, and it's important to mention the importance of supplementary nutrition. And today, we shouldn't delay this supplementary nutrition to avoid the development of allergies. I would now like to present some very interesting findings from a study, the Cognis study, where we assess two different formulas. One is an experimental formula that was supplemented with MFGM and symbiotics. And here we are going to see the effect on the development of the immune system and on infections. So this is the Cognis study. This is the design. We had 85 children on the standard formula, 85 on the experimental formula, and 50 with that were breastfed, and we look at the development of the immune system, the evolution of the fatty acids and their concentration in the gut mucosa, and we saw changes in the percentages of arachidonic acid until 18 months, and we see the differences between the standard formula and the experimental formula. And we see differences in the gut microbiota among the different groups. For example, in the experimental formula group at 6 months, we see a greater presence of lactobacillus that's more similar to that of infants that were breastfed. And the same happens with at 12 months, and at 18 months, we also see a higher level of bifidobacterium. Therefore, during the first year, the gut microbiota of the children fed with the experimental formula was more similar to the microbiota of the breastfed infants and different from those fed with the standard formula. Here we see the maturity of this gut microbiota. With breast milk, there is a slower maturation process versus the formulas, and at 18 months, this variability is not so obvious. But the experimental formula always shows to be much more similar to breast milk. This is the comparison of the two formulas versus breast milk. In green, we have the experimental formula that is more similar to breast milk at 6 months and at 12 months and at 18 months. And the functionality is more similar to that of breastfed infants, and it's different from the functionality of infants that receive the standard formula. Here in these charts, we can see many differences in the metabolic activity of the bacteria when we compare the standard formula versus breastfeeding, and then we see that the experimental formula shows a very similar effect to that of breast milk. And in terms of infections, there is a lower incidence of infections at 12 months, gut infections and respiratory infections, and fewer number of visits to the pediatrician and fewer events of fever and viral infection and fewer consultations to the pediatrician in those receiving the experimental formula versus the standard formula. And when we associate the gut bacteria with these events, we see that at 6 months, there is bacteroiditis that are associated more with vomiting and diarrhea. The same happens with proteobacteria at 18 months. It's also associated with vomiting and the higher prevalence of firmicutes protects from vomiting and diarrhea. Therefore, firmicutes seem to be less beneficial when looking at obesity. However, we see the results here with the vomiting and diarrhea. And when we look at the three groups, breast milk has a higher presence of firmicutes in the gut. The standard formula mainly shows bacteroiditis, and in the case of the experimental formula, it also promotes in the second place the growth of a gut firmicutes. So it seems, however, here firmicutes in the standard formula is very, very low. So this effect has to do with the similar function that we have seen between breast milk and the experimental formula. When we look at the specific relationship with the secretion of salivary IgA, there is a negative association with the secretion in the case of bacteroiditis. However, firmicutes do promote a higher level of salivary immunoglobulin. Therefore, by way of conclusion, we can say that the immune system development depends on healthy establishment and maturation of the gut microbiota. Additionally, early nutrition has a significant impact and determinant effect on gut microbiota establishment, maturation, and function. The experimental formula that has been enriched with MFGM and LC-PUFAs and symbiotics promotes a gut microbiota that's more similar to that observed in breastfed infants, both structurally and functionally, and the immune system development in this case has high concentrations of salivary SIgA that seems to be associated with a higher amount of firmicutes in the gut that will protect from infections, and there's a reduced gap between formula-fed and breastfed infants, although we need more trials to confirm this data. Thank you very much for your attention. I hope you continue enjoying this event. Thank you.
Laboratorios Ordesa y a la Cátedra Ordesa.
Hello, I would like to thank Laboratorios Ordesa and its cátedra for inviting me to take part in this 7th Scientific Symposium and focus on such an interesting topic such as the introduction of complementary early nutrition and reprogramming of health and disease. I've been asked to speak about the introduction of complementary feeding and immunomodulation. That would be the topic of my talk. Sorry. The previous speaker told us about the development of immunity throughout the first life stages of the person, of the individual. And we know for sure the reasons why the feeding habits have to cease to be just based on breast milk and to incorporate other foodstuffs. So the when, what, and why are well known to all of us, but not so well known is how to justify the introduction of these foodstuffs in the development of immunity and the development of pathologies related to it. So the effect of complementary feeding over the health conditions the growth and an adequate bodily composition. We also know that it makes an impact on neurodevelopment, although to a lesser extent than breastfeeding, and also in the establishment of gut microbiota. Maybe we have a lesser knowledge or we have more doubt about the role played by complementary feeding in the development of the immune system, more specifically in relation to development of infections, allergia, different manifestations of allergia, and immune disease such as celiac disease and diabetes. There is no evidence, or at least we have no data, to allow us to relate that the introduction of complementary feeding, the specific timing or the specific composition of complementary feeding are associated to the risk of suffering infection. So we can summarize it by saying that nor the timing nor the type of foodstuffs can have a direct relationship with the exception of food safety. Now I would like to speak about the introduction relationship or the link between allergies and complementary feeding, because our body recognizes new antigens that are introduced by the diet through a more or less complex mechanism of exposure and the presentation of those antigens in different cells, so as to finally develop a family of regulatory T lymphocytes in the lamina propria, and some of them also go to the systemic blood torrent, and they finally end up allowing tolerance. So this exposure to antigens somehow justifies the fact that we are capable of tolerating foreign antigens that we take in that end up in our bodies through feeding. So the next question would be, why do some individuals develop allergic manifestations? And second question derived from that, is there a direct link between food antigens and the possibility to develop allergies, any type of allergies? What would be the ideal timing for introducing complementary feeding, and whether there are differences according to types of foodstuffs? Regarding allergies, so I insist on the question, what would be the relationship between the timing and the moment in which we introduce complementary feeding and the onset of food allergies, etc. So there is still a great deal to, a lot to research to find out whether some people develop allergies, whereas others don't. In the year 2019, a new systematic review and meta-analysis was published in which they tried to give an answer to these two questions: relationship between the timing of the complementary feeding and the onset of food allergies, atopic dermatitis, asthma, and allergic rhinitis. And the second question, in this article or in this presentation, I will remark the most interesting aspects. First question: relationship between the timing of the complementary feeding and onset of allergies. So the difference is between an early timing, early moment, 3-4 months of life, against or compared to late introduction at 6 months. So there were 31 studies, and regarding food allergies, they found a higher level of evidence. The outcomes, some of them found a direct relationships, others didn't. So it appears that introducing complementary feeding over 4 months, appears to increase the risk of suffering allergies. Regarding the second question, that referred to the type of quantity of foodstuffs. At that time, the review included 39 studies, focused mostly on those foodstuffs that tend to be more allergenic, that is peanuts, other types of nuts, eggs, fish, shellfish, dairy products, wheat, and soya, whereas compared to other less allergenic foodstuffs. Let's start with peanuts, tree nuts, and seeds, because we have more evidence. So results from 14 studies were collected, 9 of them geared towards answering to the question related to the introduction of this foodstuff, the peanut, during the first year of life and the onset of food allergies. So these studies state that early introduction during the first year compared to a late introduction diminishes the risk of allergy to peanuts, and there is less evidence to other tree nuts and seeds. And for the other allergic manifestations, no evidences were found. Sorry for the mistake regarding eggs and fish. There is moderate evidence that the introduction of eggs in the first years of life, and from 4 months onwards, diminishes the risk of egg allergy, and there is a limited evidence with respect to fish. So there is some evidence that the early introduction of egg facilitates tolerance, whereas with fishes there are some evidences, but not so strong. Regarding dairy products, wheat and soya, number of studies is important, but in none of these 3 groups of foodstuffs we were able to demonstrate that the early introductions, around 4 months, lead to a decrease of manifestations, allergic manifestations, be it respiratory, food, or skin allergies. In this slide, I have shown some of the data of the lead study. This study focused on trying to demonstrate that the early administration during the first year of peanuts in children with a high risk of suffering peanut allergy induces tolerance compared to a late introduction. In this group, we randomized nearly 630 infants, which were subdivided in 2 different groups. The first group was a high risk of suffering allergy. Those children presented severe eczema or egg allergy. So this subgroup was subjected to an allergy test, and they were subdivided into groups: positive skin tests and negative skin tests. And in turn, they divided it into a group of children which were introduced to peanuts between 4 and 12 months, and the second group when introduction was a late one, over 3 years of age. So in the follow-up of the total cohort, as you can see on the right-hand side, the group that was with early introduction of peanut during the first year was 5 times less likely to suffer food allergies compared to the late introduction group, and results were positive both for those children that had that presented negative and positive skin allergies. So it makes a difference in the early exposure to an allergenic foodstuff such as peanut, a very common type of food in some cultures. So it's been proven that the early introduction between 4 and 12 months diminishes in a significant way the risk of suffering egg allergy. When we try to collect, I try to collect the outcomes, the results of the previous studies, we can say that regarding peanuts, introduction between 4 and 11 months diminishes the risk of suffering allergy with a high level of evidence. Regarding egg, with the same results, but with a slightly higher level of evidence, whereas for other groups of foodstuffs, milk, wheat, fish, soya, there are no strong enough data so as to be able to assert or to assign a higher or a lesser risk, depending on the timing on when this food is introduced in the diet of the child. These results led to some scientific associations or societies to modify guidelines, recommendations of introduction in specifically about peanut, and they do it depending on the risk of suffering allergy to this foodstuff. As you can see, this slide belongs to the National Institute of Allergy and Infectious Diseases in the United States. Same as the Australian Society, they have divided them in 3 groups. The first one, these children had a high risk of developing allergies, so early introduction is the main guideline, the main indication to induce tolerance and therefore to prevent the onset of food allergies. In lower risk groups, second and third group, the guidelines would be the normal ones, the standard ones. The rationale, the justification of this connection is not well known. That microflora could play a role, as I've highlighted here. We all know about this. This is quite well known. The development of the microbiota starts from the fetus to through infancy to childhood, and also the child feeding habits plays a very important role. But this moment of cessation of breast milk feeding seems to play a very prominent role. There are clear differences in the microbiota of children with allergies. That's compared to those that don't have any allergy, but we still need to find out more the differences in the composition of microbiota. Maybe there are other factors that justify those fundings. So there is room for study. Regarding the moment, the timing of introduction of gluten and the risk of having celiac disease, and I will also speak about the development of diabetes mellitus type 1. As you all know, in celiac disease, there is probably an increase in gut permeability that favors that some components in the gluten activate the immune system, thereby producing an autoimmune reaction against cells present in the gut mucosa. In recent years, there has been a lot of debate about the protecting factor of breastfeeding against the development of celiac disease in individuals at risk, but the data from the PREVENT studies and the Italian study confirmed that there was no such protecting role. Therefore, there is still a certain degree of doubt or uncertainty about what to do about the introduction of gluten to avoid the risk of developing celiac disease. And I highlight the recommendation given in 2016 by the European Society of Nutrition. So it recommends the introduction between 4 and 12 without specifying which is the ideal quantity, and it doesn't specify whether it should be introduced earlier in individuals with higher risk, having siblings with the same disease. EAT study, this is a study about early exposure to 6 groups of allergenic foodstuffs, including wheat. And in this EAT study, in the sub-study that related to intake of gluten and development of celiac disease, the main conclusions of this study with 1,000 infants, early introduction 4 months of high quantities of gluten diminishes the risk of developing celiac disease in the general population. This study was quite controversial, so right now we cannot recommend, can make a firm or final recommendation regarding this study or other ongoing studies. Regarding the diabetes type 1, there is an asymptomatic stage, then presymptomatic and symptomatic stage. There has always been a certain degree of controversy regarding the role played by feeding habits. Of course, breastfeeding seems to play a protective role, and some dietary factors could have some predisposing effect to certain proteins in the milk. But at this moment, there are no concluding clear studies that specify which is the role played by antigens present in the diet regarding the possibility or the likelihood of developing diabetes mellitus type 1. Some considerations that to take into account, some take-home messages, introduction regarding the introduction of complementary feeding. This timing is being delayed because the WHO recommends to maintain exclusive breastfeeding until the 6 months. So there is a progressive trend towards a later introduction and also baby-led weaning or introduction of, sometimes fish and meat are introduced on later on. So when we analyze the content of LC-PUFAs in the diet of infants over 6 months, we see that intakes are lower than the recommendations, and in part it has to do with these new ways of introducing foodstuffs, complementary feeding. In this symposium, you will have the opportunity to hear Professor Calder. He will speak about LC-PUFAs, and he might be able to give some more details about these findings. These are data from La Salma study with more than 1,000 infants. So those babies and infants, regarding their intakes of vitamin D, are lower than recommended. We know the role played by vitamin D, LC-PUFAs, and how they impact in the maturation of the immune system. My last slide. I'm going to try to cover final messages. So this, as a way of summary, exposure to diet antigens is a key point in the induction to tolerance, hence the importance of complementary feeding regarding the maturation of the immune system. When the timing is early, there seems to be evidence that these conditions, the onset of allergies, and regarding some foodstuffs, peanuts and eggs, early introduction, 4 months onwards in individuals at risk, diminishes the likelihood of developing allergies, whereas this hasn't been proven for low risk or no risk infants, not for many foodstuffs. Regarding gluten, at this very moment, we cannot specify which is the best timing, the best moment in which to introduce gluten, nor in which quantities, so as to diminish the risk of suffering celiac disease in individuals with greater genetic susceptibility. And last but not least, some changes in the guidelines regarding complementary feeding imply important changes in the specific nutrient intake whose influence, long-term influence on our health, we don't know. And my final message, of course, is a word of caution. Let's be prudent when introducing complementary feeding, not only in terms of tolerance, induction to tolerance, but also in other areas. Therefore, it is absolutely indispensable to support modifications in complementary feeding when data from well-designed scientific studies allow us to give transfer this new confidence on the population so as to avoid confusion. Thank you for your attention. I'll be glad to answer to any of your questions.
I'm going to talk about the role of long-chain polyunsaturated fatty acids in the development of the immune system. The immune system is the system that protects us against pathogens, that is harmful bacteria, viruses, and so on. It's a cellular system, but the cells are also congregated together in different tissues, like the spleen and the lymph nodes. We know that a well-functioning immune system is key to providing good defense against pathogens. We know that because people who are immunocompromised or immunosuppressed, so with weakened immune systems, have increased susceptibility to infections and to infections becoming more severe, even fatal. Essentially, the immune system works by distinguishing self from non-self, that is, things that belong from things that don't belong. But of course, every day we eat non-self, we eat plants, we eat animals. Also, our GI tract, our mucosal surfaces, our skin are covered in micro and macroorganisms. So we have chronic exposure to non-self. And it wouldn't make a lot of sense to mount vigorous immune responses to the things we eat or any harmless micro or macroorganisms that we find on us or in us. Therefore, as well as providing defense against pathogens, our immune system provides tolerance against non-threatening organisms, to food components, and to ourselves. So if you think about this, the immune system has a balancing act. It has to balance up its capability to defend us against threatening organisms with its capability to tolerate ourselves, our food, and non-threatening organisms. Breakdown in this balance, so a loss of host defense, for example, increases susceptibility to infection, whilst a breakdown in tolerance increases the likelihood of immune-mediated diseases, because, of course, we react to ourselves, to our food, and to things that are normally harmless. So this dysfunctional imbalance can lead to disease, and that disease can be severe in some cases. Now, if we think about early life, of course, our immune system is very immature when we're born. We have immunity before we're born, mainly through the passive transfer of antibodies across the placenta from the mother. When babies are born, they have immune cells present, they have immune organs like the spleen and lymph nodes, but the cells are immature and don't function very well. So over the course of weeks to months to years, the immune system has to mature. So we have to develop the capability to mount appropriate immune responses and to mount tolerogenic responses. Interestingly, this immune maturation goes hand in hand with the maturation of our gastrointestinal tract and its colonization with bacteria. What I show here is the green arrow of optimum immune maturation giving a healthy immune response, and the possibility of deviations with poor immune maturation that would increase susceptibility to infections in infancy and childhood, or increase susceptibility to allergies in infancy and childhood. So we have to move from the state of passive immunity, immature immune cells, no immunological memory, and a sterile environment, to a situation of mature immune cells, balanced immunity, and an endogenous microbiota, which of course isn't sterile, with well-functioning innate and acquired immune responses. So factors influencing immune development include the microbes and their products that we're exposed to in infancy and childhood, the microbiota itself, which is acquired from the mother and from the environment, and the diet through exposure to new foods, as well as important nutrients within those foods that might be maturing factors, things like nucleotides, gangliosides, and even the fatty acids that I'm going to talk about. Of course, in early life, the diet is breast milk, which should provide factors that promote optimal immune development and optimal gastrointestinal development. So, of course, breast milk has all of these factors in it that help to support and mature the baby's immune system. Things like antibodies, immune cells themselves, cytokines and growth factors, these other complex structures like nucleotides, gangliosides, and so on, and I include the long-chain PUFAs on my list here of important immune factors in breast milk. So let's dive now into the polyunsaturated fatty acids. So we have two main families, the omega-6 family on the left and the omega-3 family on the right. The simplest forms of these families are linoleic and alpha-linolenic acids, but the bioactive forms are the more complex arachidonic acid on the omega-6 side and EPA and DHA on the omega-3 side. And you'll see that arachidonic acid, EPA and DHA all give rise to bioactive lipid mediators, the eicosanoids like prostaglandins and leukotrienes, and the pro-resolving mediators like resolvins, protectins, and muresins. And often the members of the omega-6 side oppose the action of the members on the omega-3 side and vice versa. So there's mutual inhibition of metabolism in these pathways, and there's mutual antagonism of the biological activities of mediators formed from these pathways. So once again, I think we need another balancing act here, a balance between the omega-6 and omega-3 fatty acids. Now, how can these fatty acids influence immune maturation and the immune response? Well, long-chain fatty acids are incorporated into cell membranes. Once they're in the membrane, they influence the physical nature of the membrane, its fluidity. They influence signaling activities that happen within membranes, for example, the formation of lipid rafts, which are signaling platforms, and they influence the production of the second messengers, the lipid second messengers, things like the eicosanoids and the pro-resolving mediators that I've already mentioned. So through these effects on membrane structure and function, long-chain polyunsaturated fatty acids influence signals that cells are producing, and those signals influence gene expression, protein production and secretion, and cellular activation. Through these mechanisms, long-chain PUFAs can influence the immune response, including its inflammatory component. So let's now start looking at trials in humans that are relevant to the mechanisms that I just mentioned. So I'm going to show you a series of trials from preterm infants to term infants and then in pregnant women where long-chain PUFAs have been used as an intervention, and the outcome is something to do with immunity, inflammation, infectious disease, or allergic disease. So the first trial is the study of Catherine Fields in preterm infants who were supported with human milk or formula with long-chain PUFAs for 42 days. The long-chain PUFAs were a mix of arachidonic acid from the omega-6 side and DHA from the omega-3 side. The researchers monitored immune cell numbers and immune cell responses in the blood of those preterm infants. And essentially, they found that infants reared on human milk or on formula had different trajectories of these immune outcomes, but when long-chain PUFAs were added to the formula, the trajectory was very like that seen in human milk and was different from that seen in formula without the long-chain PUFAs. So in other words, adding long-chain PUFAs to the formula results in immune effects that are consistent with those seen in human milk. This is another study by the same group, this time in term infants, again comparing formula to formula with long-chain PUFAs, and there were breastfed comparators. These were term infants fed from age 2 to 6 weeks, again on a formula containing arachidonic acid plus DHA compared to a formula without. Again, they monitored immune cell numbers in the blood and cytokine profiles, and they found that the formula infants without long-chain PUFAs were different from the formula with long-chain PUFAs, but the formula with long-chain PUFAs was similar to the breastfed group. So again, compared with formula-fed infants, the formula plus long-chain PUFA infants had an immune cell distribution and cytokine profile that wasn't different from that of the breastfed infants. So these are two studies, one in preterm, the other in term infants, suggesting that adding long-chain PUFAs to formula creates an immune maturation, an immune profile that's more like that achieved with breastfeeding and less like that achieved with formula without the long-chain PUFAs. So this would suggest that long-chain PUFAs in breast milk are very important as far as the immune system is concerned in preterm and term infants. Now, this is a study a little bit different from those ones. It's a large study in term infants, and the infants were randomized to receive fish oil or placebo in addition to being breastfed. The dose of EPA and DHA is shown here. So this was an omega-3 intervention. Arachidonic acid wasn't involved, and the period of intervention was the first 6 months of age. So many immune outcomes were not different between the groups, but there were some differences. For example, in the fish oil group, the interleukin-13 response to house dust mite was lower than in the control group, but the cytokine production in response to a T-cell stimulant was higher in the fish oil group. So also, higher DHA status was associated with a poorer or a weaker response to allergens. So the conclusion from this study was that omega-3 fatty acids in early life have immunomodulatory properties that are allergy protective. Study from Danish researchers, now in infants aged 9 months, and for 3 months they were given cow's milk or infant formula, either with or without fish oil. So this is a 2x2 factorial design. Higher dose of omega-3 fatty acids now, getting up to about 900 milligrams per day. Again, some outcomes were not affected by the omega-3 intervention, but fish oil increased interferon gamma production when cells were stimulated with this bacterium. And the authors concluded this suggested that fish oil resulted in better immune maturation. So a rather consistent pattern coming out from preterm infants, newborn infants, and infants beyond 6 to 9 months of age. So the idea here is that optimizing long-chain PUFA supply gives optimized long-chain PUFA status in stores in the infants, and this will optimize immune development and function, and this should give good defense against pathogens and prevention of allergies and other immune disorders. So what we've seen so far, certainly the first part of this causal chain is supported by the research I've shown. So let's now look at studies looking at long-chain PUFA exposures early in life and clinical outcomes a little later on. So this is a study where data from 2 randomized controlled trials in term infants were combined. The infants were either in a control formula or formula plus long-chain PUFA group. The long-chain PUFAs were the combination of arachidonic acid and DHA. They received the formulas from very early in life up to 1 year of age, and then they were followed up for three years. And these are the outcomes, the clinical outcomes at 3 years, expressed as odds ratio for the long-chain PUFA group relative to the control group. And the outcomes are wheezing and asthma, atopic dermatitis, the combination of wheezing or asthma and atopic dermatitis, any allergy, and upper respiratory infection. And you see the odds ratios are much below 1. In fact, there's a reduction in risk of 60 to 80% in all of these outcomes, and they're all statistically significant, as you see, apart from atopic dermatitis, which is approaching significance. So this is a suggestion that long-chain PUFAs early in life protect against respiratory disease and maybe atopy in the first 3 years of life. Similar sort of study, term infants in 2 groups, control or long-chain PUFAs received from 1 month from 1 month of age with follow-up at 1 year. Again, the combination of arachidonic acid and DHA. The outcome here is bronchitis or bronchiolitis. These are the proportion of infants at each age showing the outcome, and you'll see the outcome is always lower in the long-chain PUFA group, significantly so at 5, 7, and 9 months of age. So a suggestion that early long-chain PUFAs protect again against respiratory illness. This is data from a study of Susan Carlson's, where infants were fed formula without long-chain PUFAs or with long-chain PUFAs, but at different ratios of arachidonic acid to DHA, and then they were followed for 4 years, so beyond the period of formula feeding, and what they showed was allergic disease was lower in the first year in infants receiving long-chain PUFAs than control, and time to first allergic illness and to skin allergic illness was longer in the long-chain PUFA group than the control group, and there was also a trend to reduced wheeze and asthma. So rather consistent findings across these studies of either the combination of arachidonic acid and DHA or of omega-3 fatty acids themselves. Again, going back to the study of term infants randomized to fish oil or placebo for the first 6 months of life, and then looking at allergic outcomes at 12 months of age, no overall effect of fish oil, except it was found that infants with higher omega-3 status at 6 months had less eczema at 12 months. So I think maybe even though this is quite a large study, the sample size wasn't enough to actually demonstrate the effect of fish oil when the two groups were compared that came out when the status of omega-3 fatty acids was investigated. And that might be because these infants were also all being breastfed and would have been receiving different amounts of omega-3s through breast milk. Let me quickly now turn to studies in pregnancy. So the studies I've talked about so far were in newborn infants or young infants. There are studies in pregnancy addressing this question as well. So the real classic study is the study of Susan Prescott, where allergic women were randomized in pregnancy to receive placebo or fish oil from week 20 until delivery, a very high dose of omega-3 fatty acids, and many immune outcomes in the cord blood, and then clinical outcomes at 1 year of age in the offspring. So in the cord blood, the pro-allergic cytokine interleukin-13 was lower in the fish oil group than the control group, and there were also some different blood cytokine responses between the fish oil and the placebo group, with fish oil reducing the effect to some of the response to some of the allergens. But the really interesting thing from this study was that infants in the fish oil group had a lower risk of severe atopic dermatitis at age 1 year, and they were significantly less likely to be sensitized to egg at 1 year of age. So intervention with omega-3s in pregnancy seemed to be reducing the risk of some allergic outcomes in the babies at 1 year of age. Similar findings from a Swedish study where, again, pregnant women were randomized to fish oil or placebo during pregnancy and lactation, and the offspring were studied at 6 and 12 months of age. And here you see the outcomes at 12 months of age in the placebo group in green and the fish oil group in yellow, showing skin prick test positivity to overall, sensitization to egg, having allergic eczema, and having food allergy were all significantly lower in the babies of mothers who took omega-3 fatty acids compared to the babies of mothers who took placebo. These sorts of studies were put into a meta-analysis by Maria Macready's group published in 2016, and this is a table actually from a review that Liz Miles and I wrote, but which repeats the data from or summarizes the data from the BEST meta-analysis. Essentially, the meta-analysis shows that omega-3 fatty acids in pregnancy reduce atopic eczema, reduce IgE-mediated disease, reduce skin prick test positivity to any allergen, to hen's egg, and to any food extract in the first 12 months of life. Now, more recently, we've had this landmark study published in New England Journal of Medicine, where a large number of women were randomized to placebo or fish oil from week 24 of pregnancy until giving birth. with 2.4 grams of EPA plus DHA being delivered. And then the children were followed up to 5 years of age, and the main outcome was persistent wheeze or asthma. And you see a reduction of about 30% in risk of wheeze or asthma in the children once they got to 2 or 3 years of age in the omega-3 group compared to the placebo group. So children aged 2 to 5 of mothers who took fish oil during pregnancy
Hola, hola.
had a lower risk of wheeze or asthma than children of mothers who took placebo during pregnancy. Now, in a subgroup analysis, they looked at the effect of maternal omega-3 fatty acid status at the start of the study on the outcome. So on the left, we see the outcome in mothers who started with low omega-3 status, and on the right, we see the outcome in mothers who started with high omega-3 status. And you see the outcome is only seen in mothers who started with low omega-3 status. In other words, omega-3 fatty acid intervention works in women who have low omega-3 status, but it doesn't work so well in women who have high omega-3 status, which I think makes sense. So just to summarize, what I've told you is the immune system develops early in life, and nutrition is an important maturing factor. I've told you that long-chain PUFAs, arachidonic acid, EPA, and DHA all influence immunity, including the inflammatory component, through multiple interacting mechanisms. And I put it to you that a balance in the supply of omega-6 and omega-3 fatty acids is likely to be important. I went on to talk about studies in infants and in mothers, and the findings of immune markers in infants fed formula with long-chain PUFAs, arachidonic acid, and DHA are similar to those of breast milk-fed infants, but are different from those of formula-fed infants, where the formula doesn't contain long-chain PUFAs. Then I showed you that infants who received formula and long-chain PUFAs had lower risk of allergies and respiratory disease, respiratory illness, than infants who received standard formula without long-chain PUFAs. Infants who received EPA and DHA may have better immune maturation with a more Th1 and a less Th2 response, so a less pro-allergic response than those who don't receive omega-3s, so they have a less allergic phenotype. And then I went on to tell you about studies of infants of mothers who had received omega-3s in pregnancy, having lower risk of allergic disease and asthma, right out perhaps to 5 years and beyond. So as far as immune development is concerned, we need this balancing act between defending ourselves against infectious organisms and being tolerant to ourselves, to our food, and non-threatening organisms. I think the data from preterm infants, term infants, and studies in pregnant women looking at the offspring show quite clearly that long-chain omega-3 fatty acids have a very important role in setting this balance, making sure that the infant can defend itself against harmful organisms, but can tolerate things like food that give rise to allergies. Of course, some of the studies of the formulas also include the long-chain omega-6 fatty acid arachidonic acid. But it's not really clear whether arachidonic acid is necessary in those formulas or whether the effects seen are due to the DHA, the omega-3 fatty acid. So it may be that omega-6 fatty acids do play a role in immune development and in this balancing, but that needs to be demonstrated, I think, through other sorts of studies comparing the effects of these different PUFAs and of different combinations. Thanks very much for your attention.
We can hear any sound? No. Seems that the image is back. So we're ready for the Q&A round. Getting ready.
Bueno, muchas gracias a todos los
Thank you
ponentes.
so much. I would like to thank the professors and speakers, and now Professor Ascension will chair this colloquium, this Q&A round.
Ascensión.
Ascension, when you wish.
Bueno, iba a comenzar Ascensión, pero como no está...
As Ascension was the one to start with, we will try to answer the first question having to do with, in your opinion, what's your opinion about early introduction of complementary feeding in the impact on the development of allergies and diabetes mellitus? Sorry, I was just waiting in the waiting room. I couldn't access. That's why. Maybe Jose Manuel wants to break the ice. Well, ultimately, if we consider the evidence that were published with rigorous data, below 4 months, nothing, that's obvious. So from 4 months onwards, it's only been demonstrated with strong evidence, the case of peanut and eggs. So we need to be really cautious with the rest of foodstuffs. Another considerations, studies were made with a high-risk population to allergies. So we cannot make a general recommendation for the whole population. So we, at least in my opinion, we must continue with the recommendations given by European societies, exclusive breastfeeding up to 6 months, and around the 6th month introducing complementary feeding. Whereas a different situation would be those infants that have they present a high risk of suffering food allergy. Christina, what's your opinion in that respect? In any case, some people are working on studies trying to generate or to induce tolerance to different allergenic foodstuffs, not only egg and peanut, but several foodstuffs at the same time, because it appears that the immune system is capable of generating that softer response or to induce tolerance when several allergenic foodstuffs are administered at the same time, instead of waiting for a later stage, because the response will be more powerful, the immune system will be more developed, and tolerance will be more difficult to achieve. So therefore, we are trying to introduce a cocktail of allergenic foodstuffs in low doses on an earlier stage. I'm trying to generate tolerance to the higher number of foodstuffs, or allergenic foodstuffs possible in a shorter time. In fact, in the United States, some products are being sold so as to induce tolerance, and it's being sold already, in fact.
Can also join to this concept. Philip, do you hear me?
Yeah, sorry, can you repeat, please?
I was thinking about if you agree also with this concept to promote the cocktail of allergens.
Yeah, sorry, I'm a long way away from you. Yeah, I do. And I think what's come out, actually probably every speaker today has talked about this importance of educating the immune system, educating the gut microbiota. And I think if you think about what you're trying to do in that context, it's best to sort of start early, but not too early. So start at the right stage and do things in a sort of coordinated and concentrated way. So I think, introducing multiple things at this important time could be an interesting concept. And I think you're right, waiting too long until the miseducation has happened, you're now trying to recover something. So it's like you think about behavior. This is like the behavior of the immune system, and you have to manage that. So I think this is an interesting concept. I wasn't aware of those products being available, but of course we need a good evidence base before we really take these things seriously, I think.
Exactly. Yes, but anyway, it's amazing because these new concepts are changing all our thinking about the immune system during early life and during the first year. And so we have now being blocked about this idea, and I am really fascinated with this concept because I think it is perhaps it's true. This is the right way. But I don't know, as you mentioned, we need further studies to demonstrate. But yes. This is the situation. Okay.
Yeah, I mean, traditionally there was always this view that you avoided because of the danger of intolerance. You would avoid things, and that doesn't seem to make a lot of sense. It's like you're not trying to educate the immune system the right way. So I'm very pleased with the LEAP study, and actually there's another study on egg that sort of says this idea of avoidance just doesn't make sense. We need controlled introduction, I think.
Yes, this is one of the reasons why we decided to do this meeting, because and the topic of the meeting, because I think that it is really important to define all these issues, because this will implies very important recommendations that probably will reprogram some problems. And if we are able to identify clearly the timing and the type of allergens that we can give together in a period not very long, perhaps we can also go ahead with further advantages also for the gut microbiota functioning and for the prevention of many other illnesses, not only allergies or infections, but also other illnesses like obesity or other ones, because immune system is already involved in that as well. So.
Bien, es una gran discusión.
It's a great discussion, really exciting topic. But the thing is that there is a great variety of factors that intervene both in the mother and the child and the infant. So we need to take all of them into account. And up to now, studies have focused on a small part of those factors and neglected the whole picture, the whole big picture. And we need to consider all those intervening factors. One more question. To what extent can we provide a probiotic or a type of probiotic to improve microbiota and prevent autoimmune diseases? That would be the question.
Autoimmune diseases by probiotics or a type of probiotics.
I do not have data enough to say which probiotic about that, because I don't think that there are many studies with probiotics to demonstrating its effects on the prevention of autoimmune diseases. But for instance, there are some kind of studies regarding Vermex. It has been shown that when the children becomes with the hygiene theory, you have these results that in the countries where it has been applied and there are no more not too many infections with Vermex, for example, or some kind of Vermex. The problem is that the population develops autoimmune diseases. So probably that will be probiotics we need to study and design to modify the risk to develop the autoimmune diseases. Probably there is, I don't know, Philip, if you know, or Jose Manuel, if you know about some studies in this area.
Yeah, I mean, the studies I'm most familiar with are the ones that relate to allergy, which is not an autoimmune disease. It's another type of inflammatory disease. And of course, there are studies indicating that probiotics in pregnancy or in infancy, at least some studies show you can reduce allergies. I'm not aware of the data, if there is data on autoimmunity. Yeah, maybe Jose, you have some understanding there. No.
No, I'm aware of the same studies you have mentioned that are related to providing probiotics to the mother and as well as the infant once he's born or she's born, but not really regarding the autoimmune disease.
Bueno, finalmente, María Jesús, creo
Finally, María Jesús, we need to stop here because we are out of time. It's been an outstanding session, really. Somebody said, amazing session. I'm just quoting one of the attendants' ideas, thoughts. We have to stop here. Of course, there is a great deal to be discussed, to be studied, but we conclude here by thanking you all thanks for inviting us to chair this panel. We would need one more hour, at least an hour, for the Q&A round. Unfortunately, we only had 10 minutes. I congratulate you, to the panel, the organizers. So let's convene again tomorrow at 3 p.m. See you. Bye-bye.